Morning. Good morning, dear participants, and welcome to the fifth day of this training program, which was conducted in collaborative mode with managed by ICAR-NRIIPM related to the IPM and for sustainable agriculture. Uh, Regarding the recap of the last day, we have a few very important sessions and very important and elaborative lectures from the eminent scientists. The first lecture was related to the role of predators and parasitoids, which was dealt with three eminent scientists from NBAAR, Dr. Disha Varshney, Dr. Ankita Gupta, and Dr. Gracie, who actually told about the mass production protocols, issues related to the predators and parasitoids, and the DNA barcoding, and how we are supposed to conserve the predator and parasitoid, and of course their field applications for related to IPM and their management. That lecture was followed by a lecture from Dr. M.N. Bhatt, sir from ICAR-NRIIPM. Sir talked about the IPM in vegetable crops. It was a very important and elaborative lecture in which he talked about the diseases and pests, important diseases and pests, and their pest management strategies. Our participants really interacted very well in both the lectures, and there was a good interaction in both the lectures. That lecture was followed by the lecture on a very recent technology role of ICT and ESAP in pest surveillance and integrated pest management. That lecture was dealt by Dr. Prabhu Raj from US Raichur, Dr. Prabhu Raj sir, and he talked about the role of e-surveillance and how this ESAP technology can be used for surveillance and advisories for sending advices to farmers for the better management options and reducing the injudicious use of pesticides. And that lecture was followed by Dr. Mahantesh lecture who talked about the role of manage related to the extension and capacity building in agriculture per se, and how various programs can be taken up in the collaborative mode by different institutions in collaboration with manage to have a better reach to the various stakeholders and farming community for the better extension practices and better going taking the technology to the grassroots level. And that lecture was the last lecture yesterday was from Dr. Sehgal, who talked about the IPM in cereal crops. Actually, only the rice portion was taken up by sir. Rest wheat and maize will be taken up by sir today, in which he talked about the various issues related to IPM in rice and how we have to tackle them, how we have to take up the large scale IPM trials in rice, and then what are the constraints which the farmers or the field functionaries face in the field, and then how they are supposed to be taken care of. Yesterday's lectures and participants, we hope that with your interaction with the speaker, we believe we saw that you really interacted well and all the lectures were well perceived. But if any queries are there related to the previous lectures as well, yesterday's lecture or any other lecture which were dealt in last 4 days, you can like communicate us and you can tell us your queries in future also. So today we'll start with today's session. We'll start with a very important lecture. We have requested Dr. Shamila Roy, madam, for dealing in IPM in sugarcane crops. Madam is already there, and I request Dr. Mukesh Sehgal sir to start with the today's proceedings, please. In between, if any query is there from any participant, they can ask.
Yes, Dr. Rekha.
Shamila Roy, ma'am, please switch on your video, ma'am, please.
Okay. Sir, आगे recap session होगा आपको Dr. Reka?
Let me call ma'am. Ma'am, I can see her here, but her video is not there.
Go back. We have 5 minutes more to go.
Yes,
Meanwhile, tell the two, three participants to give their feedback during the validity section, right?
yes. Today, from 2 o'clock onwards, we will have the validatory session, and we request all the participants to interact during that session and give their feedback for better for our betterment, so that we improve further for the further training programs. If any difficulty you faced, or you feel that we should have done anything else, and we request all the participants, uh, specifically Dr. Deepak Jain sir, I can see, and Dr. Abhijit Ghoshal sir, please give your feedbacks. And all other participants, please interact during the validatory session. In the meanwhile, I'll share the feedback forms as well as the post-course evaluation forms also in hour or two. I request all the participants to please switch on your video so that we know that you are there.
Good morning, ma'am.
Morning,
Good
ma'am.
morning, Daksa. Good
Morning.
morning. How are you,
Fine, ma'am.
Mahesh? Okay.
So, can we start, ma'am?
Yes, I have to share my slides.
Yeah, yeah. Okay. Welcome, Dr. Sharmila Roy, as well as dear participants, scientists, and other people, those have joined here for today's session. And today we have a last day for this training program, and we have requested, just like you are aware, some of people are demanding mm, um, they are for a sugarcane lecture. Many people called me up, and so therefore I requested yesterday Dr. Sharmila Roy, principal scientist in Indian Institute of Sugarcane, Naklau, and we were very thankful that she agreed quickly for our request. Thank you very much, ma'am, for accepting our request. Dr. Sharmila Roy is the principal scientist at Indian Institute of Sugarcane Research. She is basically entomologist. She joined ICR in 1991 at NARM, and then she posted at Indian Institute of Grassland and Forest Research Institute, Jhansi. And then, after working for more than a decade, she got transferred, rather, she had taken a transfer to Kajin, and where her husband was was selected as a director. And then, after her husband's retirement, she uh, took a option to join the CSHI, Naklau Horticulture Research Institute, and then because of the cadre strength, she was transferred to the Indian Sugarcane Research Institute, and she is working now at present in the Sugarcane Research Institute. She has a very long biodata. She had published number of research papers, nationally and internationally, awarded with many awards. And because we know personally each other, so I can speak for a long time, but I don't take much of time, and request ma'am to have the share our experiences on the Indian and the IPM in sugarcane. Ma'am, these participants are from all over the India. Now, slowly, slowly, you'll see that the people will join. More than 100 with paper. Sometimes it come to 140. People are they are regularly joining with us. At the moment, we have 71, and so these are basically assistant professors, some of associate professor of the State Agriculture University, from ICAR scientists from Banana and other institutes, and some of them are students, and some of the one or two are the freelancer. We also have a man of bicycle who's traveling all over the country. Also, Prajapati is also here, and some students are also there, and some one two progressive farmers are also there. So they are very keen and very interested. Some of the KVK experts are also there. So means from all the category are there. This is our managed sponsored program on integrated pest management for sustainable agriculture. So I don't take much time, and request ma'am to uh, share her slide and to share her experience. Ma'am, please.
Yes. Thank you, Dr. Mukesh. Thank you. And now I will start. I have to share my slides. I'm going to share it, okay?
Yeah, ma'am, please.
Thank you for giving me the opportunity to share my ideas on this. Okay. Okay, is it visible now?
Yes, ma'am, is visible. Yeah.
Thank you. So, good morning to all participants and the organizers. I'm Sharmila Roy, as told by Dr. Mukesh. I joined this Sugarcane Institute 3 years back. Earlier, as he has told, that I have experience with the fodder research, insect pests of fodder research. Specifically, I worked on soil insects, microarthropods. But here I am working on the pest management of sugarcane. So good morning all. Before starting this, I will start with sugarcane. We know many facts, but still many things we do not share much, and with our specializations, we have knowledge. This what we say here, that sugarcane is a cash crop, as you all know, and it supports the largest agro processing industries of the India. And around 6 million farmers are engaged in the cultivation of sugarcane. So if we talk about the cultivated area of the sugarcane, it occupies around 3%. Slowly it is increasing also, and it is a gross cultivated area of India, it is occupies 3%. And it shares around 7% of total value of agricultural product. As we know that India is now has sugar sufficient. Now we have changed in policy. Now the sugar industries are allowed to convert their sugarcane juices, and they direct from sugarcane, they can produce ethanol, because government of India's understanding with the transport and this fuel agency, that they will mix around 8 to 20% of this bioethanol. So one of the major producer bioethanol recovery will be from the sugarcane. So we envisage that that sugarcane is going to, the cultivation at the sugarcane is going to be in a larger area, and more farmers will engage in it, because it is not only for the sugar and the ethanol, as we know that it is a very costly product, agro product of our country. So with this background, when we see that production, as we all know, that it is affected by the pest and diseases. So when we say that pest, so on sugarcane, it is reported that around 400 insects, including diseases, are affecting the production. So out of these 400, 288 are the insects, and around 125 diseases are affecting sugarcane production. Out of these 400, certainly all are not that important economically. Only 24 insects and the 12 diseases causing very heavy losses. And this losses goes up to the 20 and 30% in yield reduction. So we can see that if we convert this 20 to 30% in today's value of the sugarcane tonnage, then it comes into around 30 million rupees. So it is a huge loss. We have to, and our IASR Sugarcane Research Institute is devoted towards the breeding, its agronomy, and as well as the plant protection aspects. So as I was talking that insect pests, the important ones are the borers. When we talk about the insect pests, we can classify them into borers, subterranean pests, that is underground, below ground pests living in soil, and the sucking pests. So when the borer comes, that borers on this borer, that early shoot borer is internode borer, top borer, stalk borer, root borer, Plassey borer, Gurdaspur borer are very important. These 7 borers are very important borer pests. When we see that their economic importance, early shoot borer, here we onwards we will call it as ESB, or all the sugarcane entomologists known they term using short form ESB. So yield losses is reported around 22 to 30%, and when we go up to the sugar recovery, it is around 2%. And when we go for the internode borer, the losses is 34% around, and sugar recovery is from 1 to 2% to 3%. In case of top borer, it is 21% yield loss is reported. Range is quite high, 21 to 37%. And sugar loss is also affected by 0.2% to 4%, depending on the infestation level and the incidence. When we talk about the stock borer, it is 33%, and the sugar loss is 2% to 3% again. Root borer 35%, and the sugar loss is 3% around. Plassey borer and Gurdaspur borer is certainly they are usually still limited to certain pockets of India. The Plassey borer, as it is the name, is itself indicating the region, its infection incidence, and the Gurdaspur borer is also saying it is in a pocketed places, but this nowadays we are getting reports from other places like UP and Bihar. These the incidence of these insects. Those are sporadic, but still there are chances that they may get spread to the other places. So by these two borers, the losses are Gurdaspur borer losses are 5 to 15%, and the sugar loss is 0.1 to 0.8%. By the Plassey borer, the losses are 8.2 and 12.6%, the range. And the sugar, as you see, it's a very high sugar loss when Plassey borer damage is there on certain pockets. Apart from these borers, there are termites and white grub. These are the two insects which are subterranean, classified under subterranean insect pests of sugarcane. As we know, the termites, when termites are there, it is a heavy losses, 33%, and sugar recovery losses are also quite high. In case of white grub, these are in pockets, but wherever the white grub infestation is there is a huge losses are reported. Now coming to the sucking pests of sugarcane. The most important is the pyrella. The damage yield losses is around 30%, and sugar recovery is 2 to 3%. But when the attack is there, we lose many a times. The whole crop is affected. The whole area is affected. So not only by saying that 31% loss is there, it is actual impact may be quite higher when the pyrella attack is there. But it is under control in Western India. It was a real problem. Coming to the sugarcane woolly aphid, the losses, as you see here, 7 to 39%, and sugar recovery is around 4% losses there. In case of whitefly, it is very high because when incidence is there, it is very difficult to save the crop. The scale insect, this is actually applicable to all the sucking pests, because many a times they are them not only directly affecting the crop, they are the vectors. As we see that in case of aphid, they are the vector of sugarcane mosaic disease. And scale insects, similarly very high losses is there, but these damages are in the pockets. It's not a widespread, as we see in case of borers. And black bug is also a problem nowadays, and this problem again, the black bug is reported very high incidence in some pockets of India, and specifically in UP. So coming to our next slide, now it is the diseases, but we know that in sugarcane there are many diseases. We classify them as fungal disease, bacterial disease, viral disease, and the phytoplasma disease. Among the fungal diseases, red rot, as we know that once the sugarcane crop was as if that it is a synonym to the red rot. And recently we have seen one of our very good varieties, CO238. 0238 is is at the verge of collapse, or I should say it is collapsed because of the red rot infection. The disease itself is very important, but manageable at the same time. Unfortunately, we overlook, the farmers and the mill owners overlook this, that one single variety is propagated to maximum area. They do not follow that 60%, 40% ratio. It is almost 100% CO0238 in our UP and Bihar area. So this disease has become a problem. The variety is collapsed due to this red rot. Efforts are going on to manage this. Another disease was wilt. Fusarium saccharide is the positive organism here. The smut. Of course, in my coming slides, we will discuss a little bit more for these diseases. Here is only this, just introducing what are the diseases and the important diseases which we need to take care during our IPM activities or with the farmers, as the Dr. Mukesh said that we have KVK people here, and they have to handle all the crops in their areas, KVK areas. The another disease is poka boing. The interesting thing is this: once the poka boing disease was not a major disease, but it is appearing in many areas. And when we go on surveys, we find number of incidences of poka boing. So this is also going to be a future threat, and management of this at the very initial stage is needed. Bacterial disease, this is leaf scald and ratoon stunting disease, RSD, are the very important ones. And the viral disease is YLD, it is caused by virus, but as for scientists and science, we know that still there are many doubts. Some are saying it is phytoplasma originated, some are saying it is the viral origin. Studies are going on, but beyond this, for the management aspect, we have to take care of this. Another is the grassy shoot disease. So these are the few important diseases of sugarcane. So when we see that sugarcane diseases, how the incidence and their economic importance, when we see the incidence is 15 to 35%, it has increased with the particular variety, and losses are 10 to 15%. You are seeing here asterisk is put on some against some diseases, and this is because we say it is a zero tolerance disease. Why it is zero tolerance? Because when the disease is there, it is difficult to manage. So that is why the pre-management is required in these cases: red rot, GSD, and RSD. Smut and wilt, it is the similar 5 to 15% damage is there, and losses are due to 2 to 5% yield losses. Rust, it is 2.2 to 5%. Again, it is 2 to 5% yield losses are there with different diseases. So before going to management aspect, it is always required to know about the insect or the disease, so that by identifying them, we can propose or we should implement the management practices. That is why integrated management practices, we say, somewhere we need chemicals, as you all know, somewhere we need biological control measures, or somewhere before planting like culture measures or selection of seeds, etc. So if there is any question, you please may ask me in between, or I will continue with my presentation. Is it okay? Is it okay, please?
Yes, ma'am.
Okay. Then it is the early shoot borer. The adult looks like this. Adult shoot borer actually lays their eggs. All these insects now lay their eggs on near the midrib on the lower side of the leaf. The identifying character, when we see the insect, and the insect may not be visible to the farmers, but this symptoms will be visible. The symptoms, nature of damage, what we call it. So we see the dead hearts in a very young crop, that is 1 to 3 month old crop, where node formations are not there, but we can see this kind of dead hearts. When we pull this dead heart in the field, it easily comes out. And when it easily comes out, you find a very offensive smell is coming out. So with this symptoms, we can say that the crop is affected by early shoot borer. And when we see there are a number of holes near the shoot. So, and when you dissect or you take out this dead heart, it comes out the larva. You can see the larva is usually this kind of larva you can find out, and larva is dirty white with 5 dark violet longitudinal stripes. So the identification of the pest can also be done with the larva. The adults one may not see, or if they catch it in some trap, they can identify that this adult or this is a early shoot borer, Chylo-infescatulus adult, male, female, we can find out. But when we go to the field and the crop in the daytime, so we can see the larva and the symptoms. So, but these two things are important to identify the problem, the borer, which borer is affecting the crop. The next important one is the top borer. In the top borer is actually you will find that this leaf, you see there are no serially holes are there, because when leaves are closed, then this borer enters and dead hearts are developed. These dead heart, this dead heart is actually, you cannot pull it easily, and there is no offensive smell in this. The adult looks like this white moth. The eggs on leaves are also visible, and they are buff color, and they are having lots of hair present on the egg mass. The larva is also, when you dissect this or they open the skin or they dissect the skin, you will find the larva, and the larva is of white and having one red strip on its body, and not like that earlier violet strip. So they bore holes at the top of the shoot, and the crop having a bunchy top appearance, because the main shoot is damaged, so leaves are coming out and giving a bunchy appearance. And this kind of frass is also visible at the emerging hole, at the boring hole of the larva. So coming to the root borer is an interesting one. It actually bores just above the soil and the base of the shoot. And there is only one entry hole, not many holes, as we have seen in the earlier case. The adult looks like this. The damage is that dead hearts do not emit any offensive smell. Dead heart is again is there, but when the root is damaged, that it is visible through the crop that it is not growing. The plant is not growing. Coming to the stalk borer, this stalk borer is gives no parallel holes on the emerging leaves. When the leaves emerge, then when the leaves open, it comes up parallel holes on that. As you have seen in the top borer case, there is a one row, but when we go for the stalk borer, we find parallel rows of the holes. And these holes are visible because it is a stalk borer. That means cane is in this stalk level. It is not in the early stage when there is hardly 1 or 2 nodes are developed. It is in the stalk is affected, and you see that whole stem. These are the holes. We can see these holes. And in this case, interesting thing is this: in one cane we can find a number of larvae. In earlier cases, we find one larvae, or one larvae is at the infected dead heart. But here we find a number of larvae in the cane stem, and the larva is having 5 violet strips on its body. When you split open this cane, you will find a number of larvae, and the larvae are having this kind of strips on its body and having light brown head. So this is one of the identification feature of this stalk borer and its damage in the crop. And when we see that when it's a severely infested cane or the crop, the whole cane dries up, because when there are so many larvae are feeding on the soft tissues, the whole cane dries up. Coming to the internode borer, the larva is having 4 violet longitudinal strips, not the 5. So these are very minute differences. But when you go to the field and you find that these larvae, these 4 strips are clearly visible, unlike that 5 strip, the borer larva. In internode borer, what happens? These nodes become short, very short, and usually the length is not that short. And what white streaks on leaf appears like this. When leaf opens, you will find a white streak on this because this larvae scraps the green part of the leaf sheath. So this white streak appears. And another identifying character is that frass, that is in larval excreta, is pushed out from the cane. So this is also an identifying feature, and the spiral infested area like this you will find. This whole ring type infected area is visible. Coming to the placy borer, what happens is that the larvae aggregate on the top 3-5 internodes. And this kind of wet frass comes out from the borehole, and this frass is shiny red in color. The incidence of the borer can be easily recognized by the dried top. This top area is dried because it affects from the top 1, 2, 3, maximum 3 to 4 internodes. So the top gets dried up. The rest of the cane remains okay. So while going in the field, they identifying that it is a placy borer, you can see the top is dried up. Coming to the next borer, this is the Gurdaspur borer. As I have told, these are the regional. This placy borer and Gurdaspur are still they are in region specific, but the sporadic incidences we are recording in other parts of UP and some adjacent parts of this UP Bihar. This Gurdaspur borer larvae is having 2 phases. One phase is it eats up very heavily. That is we call it as a gregarious phase, and the another is the solitary phase. What is happening in gregarious phase? They feed on the cane tissues gregariously, and then they migrate and goes to the individual grains, and they go to then they enter into the solitary phase. They made entrance hole in the leaf sheath, on the leaf sheath, and the third internode. When the sheath is not there, because on the top internodes the sheath is less, and they enter through this leaf sheath mode and internode area. Yeah. The larvae, after entering into the internode, moves upwards in a spiral manner. This you see, this kind of things. They keep on forming spirals, and they move like this. Not the straight, like internode borer, stalk borer, we find no straight channel is there. But here in this case, they form the channel, split open the cane, and you will find that is a spiral is there on their feeding channel. There's a spiral, and while feeding, they make small tiny holes, and that is visible from the outside. Unsplitted cane also, you can see this kind of damage. Now coming to the sucking pest, the Spirella perpicilla, the damage is caused both by nymphs and the adults. They suck the sap of the plant. When the plant sap is sucked, naturally the leaves gets dried up. And what kind of spots they make? Yellowish white spots near the midrib they make it. It starts from near the midrib. and the population becomes, when the population becomes very high, they secrete honeydew. As we know, that is the Homopteris, these bugs, they make it, they secrete the plant sap, and on this plant sap, mold, sooty mold develops, and this sooty mold actually affects the photosynthesis of the plant, and the plant becomes blackish and brownish in color, and the uh, uh, growth of the plant and the cane is affected. In heavy infestation, this whole crop looks like black leaves, spotted yellow brown leaves, like the similar kind. I will show it in another slide. Coming to the black bug. Black bug is still restricted in Punjab, Haryana, UP, and Bihar, but their infestation is increasing. Their incidence has been reported, is now reported very frequently. So in future, this insect is also important to be taken care of by our extension workers and our KVK personals. This before taking the an epidemic or something like that, that become a major pest problem. So again, as we know that in case of sucking insects, the nymph and adult both damage the crop, unlike our borers or the lepidopteran pests, where the larva only damages the crop. And the nymph and adults are found on the leaf whorls. Leaf whorls means, I think you understand, that is the bunch of the leaves coming out from the top. And the leaf becomes yellow, brown by their damage, and the tip margins of the leaves gradually dries. Coming to the scale insects, again nymph and adults both damage, but in heavy infestation, this kind of incrustations are deposited on the internodes. This kind of deposition naturally affects the development of the cane. Cane becomes shriveled, thin, and leaves dries up. So it is, when infestation is there, the recovery of the cane is very difficult, and the yield is affected by this severely. Yeah, this photograph, if you see, almost a white pyrilla damage also looks like this crop, this whole crop, when it is under the maximum or the heavy infestation. And the whitefly also looks like this. Whitefly is just infested, looks white dotted. This is the whitefly infected leaves of the cane. And by their sucking, this whole leaf goes, and the development of cane is affected. Now sugarcane woolly aphid, the affected plants looks like this. The leaves are affected. These are the woolly aphids. This is the close picture. This is the woolly aphids are feeded by the parasites. The adults and nymphs suck the sap from the leaves, and they actually pierce the stylets through stomata. This is their beauty. Unlike this other sucking pests. White patches, when these white patches and when they join these patches on heavy infestation, the leaves becomes like this, dried up, brittle, completely dried up, and their heavy infestation, actually honeydew secretion is also takes place, and the sooty molds affects the photosynthesis activity of the plant. Coming to the subterranean pests of sugarcane, one important pest is white grub. As we know, white grub is usually found in the loose soils, not very compact soils, and they are usually in the Maharashtra. This is a real problem in Parvarana that we have noticed, and some other problems in the Western India, grain growing area of the Western India. As we know that these grubs affects the root system of the cane plants, and this kind of drying and yellowing of the crop is visible when wherever their attack is more, and they affect the whole. Of course, that not the whole cane grows out, the whole field. You can see the patches in the field. This is the adult insect. This is the grub. The characteristic of this grub is C-shaped, and this we call it as an C-shaped. And this kind of holes you will find on the shoot, or the stalk, and this is the root damage by this grub. And when you take out this plant looks like this, individual plant looks like this, drying up, and the whole cane plant you can pull the whole plant from the soil. Coming to the termites is visible very, you can identify the termite damage because that is when this heavy damage, they make their galleries on the cane itself. They when they eat the leaf, that is on a semicircular way, a semicircular manner, and it is other than what is you call confusion maybe with this grasshopper damage, but grasshopper damage as irregularly. I mean, this is very irregular way they cut the margins of the leaf. So you can identify, and another identification point is this: you will find the galleries in the field or on the plant itself, cane itself. When the heavy infestation, this whole cane is invaded by the termites, and they make their galleries in this. Entire shoot dries up. You can it can be easily pulled out, and the cane, or if some light breeze is there, and the whole cane fell down, collapses, or with the irrigation water also the cane collapses. Yeah, with this important insects which I have indicated you earlier. Now it is coming to the diseases. This red rot, it is caused by Coletotrichum fulcatum. When you split open the cane, this kind of redness you will find in the cane. When the crop is standing, this kind of red lining on the midrib. This is one of the character is visible on the standing crop, and when the heavy infestation is there, the whole plant dies up. Spindle leaves, third and fourth display drying. At a later stage, stalk becomes discolored and hollow. Later on, it becomes red. And in acerguli, this is a fruiting body. This is, we can say, hibernating portion of the red rot, fruiting body, and you will found on the rind and the nodes. After splitting open, disease stalk is when a sour smell emanates, and it looks like this. The internal tissues are reddened with intermingled transverse white spots. It is also visible here in this picture. The problem with this disease, if we say, once the disease is there, it is difficult to manage. It is better to manage before planting and while irrigation. This kind of easily, we call it, it is a easily managed disease, but difficult to control, because once the disease is there, it is not possible to control it. So in case of wilt fusarium, it's causing organism, fungi. Externally, when we see that there is a gradual yellowing and drying of foliage, shrinkage and withering of the canes. But when we see internally, as this is a split open cane is here, it is light to dark purplish or brown discoloration of the tissues we see here, and this becomes pithiness, and a characteristic boat shape drying is there. Cavity is made in the middle of the internodes. In case of a smut, this is very well visible when we go to the field. A whip-like structure is there in the field. Black color, whip-like. This is somewhat like, we call it dead heart in case of insects, but here it is in a grown grain, and they form whip-like structure, blackish color as you see. And in this whip, actually, the smut spores are enclosed. So they spread through this. And the plant itself becomes lateral sprouting. Birds start sprouting because of this, the death of the main shoot. And leaves especially, and this is a disease which happens mostly, we found it with the ratoon crops. This boring disease, which I already previously I said that this is a disease which may take a problem, which may become a problem for the sugarcane growing areas, because these diseases is again like in other diseases, if not managed at the early stage, and the crop will be affected. The early symptoms, we say it is chlorotic conditions towards the base of the young leaf. It is the leaf becomes yellowish, the base. And the base is thinner than the rest of the healthy leaves, this part of the base. In acute phases, the young spindles are killed and the entire top dries. Slowly, it gets killed and dried up. And when the case is advanced, there's a deformation of the leaves is also visible. This kind of shoot you will find. So red spikes and stripes are also developed. This is the acute stage of the disease. And for this, we suggest a management aspect before the planting and the selection of the seed, or the treatment of the seed of the sugarcane. This is another disease, grassy shoot disease, GSD we call it. It is characterized by the proliferation of vegetative buds from the base of the cane, giving rise to crowded bunch of tillers bearing narrow leaves. This kind of. The tillers bear pale yellow to completely chlorotic leaves. The problem with this, when there are so many of this, cane formation is affected. This plant remains like this, or if the cane forms, it's very small, with very few internodes are there. Cane formation rarely takes place in the affected clumps, and if formed, the canes are thin with short internodes. It has been observed that the one leafhopper is responsible for the spreading of the GST diseases in the crop. So there are vectors which are important to manage as insects to spread to stop the spread of the diseases. The leaf scald is the plant looks like this at the advanced stage. And the problem with this disease is it is a latent disease. The latent means when symptom appears, actually the plant is already seriously infected. And symptoms looks like the pencil line, very thin lines are there. And these lines are off-white with yellow borders. Later on, it becomes like this, drying of the leaves occur. This thin line, pencil lines, this is the midrib, these thin lines appear, and later on, this leaves gets dried up. So the problem with this, the symptoms appears later and the disease are already there in the plant.
Leave the second.
Yes, please.
But
Stenting disease is another disease. The presence of pinhead-like orange-colored dots of bacteria. Excuse me, may I start? So ratoon stenting disease, we see the pinhead-like orange-colored dots on the internal soft tissues in the nodal region. The affected canes showed severe stunting, as you see here in the field picture of the plant affected by this RSD. And reduced cane thickness, shrinking of internode towards top end, and external rim discoloration around the nodal region. This is the split open cane you are seeing here. Small dots are there. It is not that much clear on the photograph, but it is clearly visible when you split open the cane. The yellow leaf disease, this is the advanced stage of yellow leaf disease. The whole leaf becomes yellow. Actually, this starts with the midrib yellowing. Yellowing of the lower surface of the midrib of leaves, and this starts from the top leaves, 3 to 6 from the top. The yellowing expands out from the leaf midrib into the leaf blades as the season progresses until a general yellowing of leaves can be observed from a distance. This kind of appearance to the plant comes up, and it starts with the yellowing of the midrib. Now coming to this description, after finishing this description of the major diseases and major insect pests of sugarcane, we all know for the management or any kind of management, whether it is IPM or whatever you call chemical application or the biological control method solely or in an integrated fashion, the economic threshold level is very important. So there are, I have here made a slide of insect pests, on some insect pests, the economic threshold level on which we have to apply our chemicals. And before that, if it is a minor incidence or sporadic in a field, some few plants are affected, we may not go for it. The nature takes care of itself. In case of shoot borer, 15 to 20% dead hearts on the late variety is affecting the economic level, and 16 to 17% dead hearts for the early varieties, early sown varieties. In the case of paella, 3 to 5 individuals per leaf is important to be monitored. In case of stalk borer, 16 to 28 bored canes per row of 6 meter length is the ETL level. In case of stem borer, 17 bored internodes row of 6 meter length. That is why it is important when we go for this ETL level, you see that symptoms are important here to measure the economic threshold level at the farmer's field or at the cane growing fields of any field. Top borers, 15 to 22% incidence, and in case of white grub, 14 to 15 beetles per host tree, and rodents, in case of rodents, 13 to 15 live burrow per hectare. This white grub here, it is said as 14 to 15 beetles host tree. Actually, the white grub adults lives on the trees, adjacent trees like neem trees, shisham trees, and when we jerk the tree and they collect the beetles, it is that if they are more in number than this, we need to see that adjacent areas or the application of chemicals and other measures are required. Here it is not saying that in the cane field a number of grubs or important. Here it is the host tree, which actually lays whole beetles lay egg in the soil, and from there the damage starts to the root region of the crop. In the integrated pest management scheduling, in case of sugarcane, we have to start it before planting. We have to take care during planting, and again when the crop is growing during monsoon and during post-monsoon, that is near the harvest time. So before planting, what we have to do is certainly it is the selection of seed. Seed is very important because many diseases are in the seeds, reported seed-borne diseases like red rot, and we have seen it. Seeds should not be selected from the fields having low level of disease infection and insect pest. So there are chances some of the seeds may be damaged, if not all. So it is better to select seed from a very good field, healthy field, and before planting, that seed cane should be checked, and the sets should be treated before that. Set infested disease borers should be discarded. And at the time of planting, the major problem comes with the termite, shoot borer, and the root borer. And in cases where there are white grubs are there. So seed treatment with imidacloprid or this furrow treatment, both can be applied in case of either it is cane sets to be treated or in the furrow application with imidacloprid or chlorantraniliprole or fipronil maybe. So this is the during planting we have to take care this measure. For red rot, smut, grassy shoot, ratoon and leaf scald and other diseases, it is better to use resistant and moderately resistant varieties. The seed sets showing reddening or the cut ends decay ends, and this should be discarded. This could be a symptom for the disease. Healthy sets should be planted. Such set must be produced from crop raised from treatment of seed cane. Yes, another thing is this: seed can be treated, or these crop raised through moist hot air treatment of the seed before planting, and from there the seed crop is raised, and from this seed crop the seed has to be planted. And this actually MH80 unit we call it, and they treat the seed at 44 degrees centigrade at 95 to 99% are relative humidity for 2.5 hours. And this actually eradicates the seed-borne infection for RSD, GSD, smut, leaf scald, and red rot. And the data says that red rot can be controlled up to 80%. And application of azoxystrobin and along with this difenoconazole with this 1 ml per liter water against red rot, smut, and cluster. This is the soil treatment. We should do it at the time of planting as an integrated management of the sugarcane diseases and insects. So this is MH80 unit, and in this treatments are done for this. This is the internal view of the MH80. The canes are placed like this in the plates, and it is closed, and this kind of thing. This is a hot air treatment, moist hot air treatment. And during monsoon, after planting, this is important for the disease management. If we find in field some diseased plants, it is better to destroy those plants. Bunding of the affected field should be done to avoid movement of rain or flood water to healthy fields. And for this carriers, insect vectors, yellow sticky traps may be used for aphid vector and for the leaf hoppers. And if we found, then it has to be that insects and the sticky traps, if insects are trapped, and then it is not the question of ETL here, as you all know, that one vector is more than enough to spread the disease. So immediately we have to start with the control measures, chemical control measures. So this mechanical and cultural control during the monsoon period is very important for borers. The egg mass has to be removed, though it is really unpractical, but still it helps to control the disease. Remove the egg masses and cut the infested shoots from the ground level. Avoid irrigation at the peak period of emergence of top borer moth. Monitoring and mass trapping of adults through pheromone traps is very important. For monitoring, 4 to 6 traps per hectare is enough, and for mass trapping of the adults, for inhibiting them from egg laying, 6 to 20 traps per hectare is required. In case of white grub, where their white grub infestation is there, jarring and checking of host trees and killing of beetles is important because it reduces the egg laying intensity of the insect. Insect trap, we in IISR we have developed a combo insect trap which is sold to the many two firms, and the report is wonderful, and we are getting very good reports, and they are able to control the young beetles and helping us to reduce the white grub infestation in Maharashtra area very effectively. Deploying for the exposure and killing of grubs in the field. Crop rotation with paddy, certainly the crop rotation helps to reduce the pest population in the field, and with paddy it is found effective. Do not use partially decomposed organic matters in the field. This is not only applicable with the white grub, this is equally applicable for the termite infestation in the fields to reduce the infestation of these subterranean pests. Coming to the pyrilla, the mechanical and cultural controls, destruction of lowermost leaves which are bearing egg masses during April and May. It is quite effective. In case of whitefly, the whitefly actually proliferates with the water stress conditions. So it is important to manage water, and neither it should be dry nor it should be waterlogged. And the overuse of nitrogen fertilizers. This nitrogen fertilizer makes the plant susceptible not only to the whitefly. We have observed and have find out that the borer attacks are also enhanced when more excessive nitrogen fertilizers are used. So it is better to restrict oneself to for the recommended dose of nitrogen fertilizer. Detrashing and destruction of leaves bearing whitefly puparia before the emergence of the adults, determined by the presence of T-shaped split in the dorsum of the puparia. So it is better to detrash the leaves where the puparia is there. So in IPM, it is we say how it is generally recommended not to use chemicals, but when there are heavy infestation, chemicals are the only options left with. So in case of ESB, chlorantraniliprole is very effective, and if chlorantraniliprole is, or it can be, if two sprays are there, we can alternate it with chlorpyrifos or fipronil. It is always advisable that the same insecticide is not to be sprayed on and on. So it has to be rotated with some of the other insecticides. In case of top borer, carbofuron or chlorantraniliprole in the root zone application is effective up to the third brood, means third generation of the borer, it is effective. In case of stalk borer, chlorpyrifos and monoprotophos are recommended. In case of black bug, imidacloprid or monoprotophos. In case of white grub, fipronil along with imidacloprid is giving effective control. In case of termite, bifenthrin, chlorantraniliprole, etc. are giving good control. And Pyrilla, we recommend we don't go for pesticides. In Pyrilla fields, we have observed there are a number of biocontrol agents. They control the pest, and during monsoon, the build-up of Pyrilla population and the build-up of their biological control agents are simultaneous. Hence, very soon this Pyrilla are controlled by the biocontrol agent. But still, if it is required, the chlorpyrifos and monoprotophos are recommended. And these are all recommended through CIRBC. And this website is there. From this, actually these are the only recommended pesticides, though our vendors sell many pesticides, but it is better, it should be suggested by the KVK and our extension people that recommended pesticides is always better to use. And if you visit this website, you can find pesticides usage for different crops and along with the sugarcane. Recommended pesticides. So coming to the biological control, we know that biological control solely depends on the trophic interaction of the pest versus biocontrol agent. So it is a 3-level tritrophic interaction. The first trophic is the sugarcane, then it comes the pest, second trophic level, and their predators or biological parasites, parasitoids, predators, whatever is you call it. It comes to the third level of the trophic level. So in an ecosystem, what we call in IPM, if we want to manage, then we have to manage the ecosystem. The habitat management, the habitat engineering words are coming out very frequently. Even nowadays, we are calling it as a natural farming, not natural farming in the terms of using only the cow dung or cow urine, etc., etc. It is the natural farming is imitating the nature and its components. That is their trophic levels to be promoted, to be protected, to be strengthened. Till date, what we do in biological control, we inundate the biological control agent to the affected site for the particular pest or particular diseases. We do it. But with the coming threats to the environment and to the human health, it is always advisable to protect the environment, the ecosystem. The ecosystem is not only the crop, the ecosystem is actually adjacent to the crop. What is growing the nearby field of the sugarcane, nearby what are the weeds or the wild plants, wild trees or the trees available in surrounding, how they are useful in protecting the biocontrol agents and the predators. Actually, what happens when when the biocontrol agents do not have their alternative host, their environment, their habitat, after the crop is removed and the pest is not there, so their population dwindles. When their dwindling population takes time to build up, by that time pests like weeds having the nature to multiply faster than their biocontrol agents. So it is the habitat engineering comes in picture, planting plant crop which may affect as a trap crop, which may affect as a only the sheltering crop, providing food and the coming generation when the pest is not there, the main host is not there, but other host is there and their food is available. So it is a point of research nowadays how we protect in the name of IPM the surrounding. It is not the integration of the methods, it is the integration of the environment and the ecosystem and the adjacent plants to protect our crop. When the sugarcane planting area will increase, certainly the pest problem will going to increase. And for production, we are forced to have new varieties, which usually hybrid varieties are susceptible to the pests and diseases. For this, it is becomes day by day important to protect our habitat and modify our habitat to protect our main crop. So it is another point of discussion. Here I do not have this kind of presentation, but yes, the work is required on this. And so here you see that some asterisks are there. These are actually most exploited natural enemies of sugarcane pest. So in biological control, there are some recommendations by our fellow scientists. For ESB, Trichogramma calonis is advised, and for top borer, Isotoma javensis, 125 females per hectare, gravid females, and Trichogramma japonicum. Like this, you will find the recommendations in textbook or also in the recommendations from the IISR booklets and brochures. And for other borers, here it is root borer, stalk borer, internode borer, Gurdaspur borer, glassy borer, like this. Trichogramma calonis is found effective, and the Cotacia flavipus. For pyrilla, I was talking that this is one of the very good example of biological control, because in nature, they both the host and the parasitoid, they multiply simultaneously. So if you have to release, then Epicrania melanocliuca, 4,000 to 5,000 cocoons may be released, and the another fungi Metarrhizium is also effective against the pyrilla. In case of woolly aphid, Diphaphidifora was found effective and had giving good control. In case of black bug, Beauveria bassiana was found effective. White grub, again the Beauveria bassiana. This white grub is actually the all these entomopathogenic fungi, and some nematodes are also reported controlling the white grub. The scale insects, we found it that Chiloporus negrita are quite effective. Whitefly, we find nymphal pupal parasitoid is very effective, and redistribution is also important, because in places where they are naturally infecting white flies, from there they can be transferred or transferred to the affected fields. So now in the end, before integrated pest management during post-harvest, post-monsoon, that is up to the harvest period, detrashing and dry leaves at the monthly interval gives good control, and removal of water shoots. In sugarcane, what happens when it is affected by some pests, some water shoots, the side shoots come out from the plant. If you remove them, then the borer population gets reduced. And this is good for even for the ratoon crop also. The population built-up is not there for the borers specifically. And when we harvest the crop, it is advisable to harvest quite deeply, and the trashes are to be removed, extra trashes. Ratooning of diseased crops should be avoided. Crop rotation should be followed in the severely affected fields. The crop rotation, which is advisable to not when they like no pink borer, now it is a problem, but when we rotate the crop, sugarcane crop with the rice, pink borer becomes a problem to the sugarcane crop. So rotation crop is also to be is very important to see, like no army worm. It affects our ratoon sugarcane crops when there are adjacent wheat fields are there. Wheat is harvested and the armyworm migrates to the sugarcane and affects the ratoon crop very badly. There are reports from our UP, many places where near Shahjahanpur like this, we have find this problem. So rotation of crop is important, and the selection of crop for rotation is also important. So thank you. It is open for discussion.
Thank you very much, ma'am. Now it's open for discussion. Somebody have a question, query, or clarification? Please, participants, please.
Good morning, ma'am. Good morning, ma'am.
Yes, please.
Ma'am, I have a doubt regarding coca boying, ma'am. Whether which stage of the crop is susceptible for coca boying, and what are the conditions that predisposes the crop to this disease, ma'am?
Actually, we have observed this poka boing in the ratoon crops mainly. And another thing we have find it out, the affected crop, if it is nearby some insect problem like this aphid and this plant hoppers disease are there, then we find it is affecting poka boing. But it's not like that any conditions is important, weather conditions or something. Is a viral disease mainly, as you have seen, or yeah, there is a controversy. We are facing this controversy.
Yes, ma'am.
Somebody is saying it is phytoplasma oriented, and somebody is saying viral. We
I
have started
understand
working.
that. I understand it is due to fusarium, ma'am. But whenever we visited the field, whenever there was an incidence of this disease, we were able to, as you said, we were able to find leaf hoppers and also mealybugs, ma'am. So whether there is any association
What is
or
leaf? What you said, leaf? What?
Phyrella, ma'am. We have seen phyrella. We have seen also
Phyrella.
mealybugs
Okay.
associated with the disease. So whether it is because of fusarium or is it a complex? That is what I want to ask.
See, it is actually the basic fundamental of this. If you go, these sucking insects, it could be any aphid or jacids, or you are saying that you have seen it with the pyrella infected fields, isn't it? So it is the sucking insect. Actually, whether it is, then it is will be transmitted to the insects with their saliva and their mouth parts. That is happening. Of course, we
Yes,
have not come through this pyrilla infested fields with the poka boing.
yes.
That is true. We have find some aphids, we have find some deserts. That one name is I
Do
have
we have
given. Aphids, we have not confirmed it yet, so I have not put the name
Madam,
there.
in some cases, there is no insects also, but you see the disease in sugarcane. Hello?
Ah, yes, your voice is very low. No, I can't hear you properly.
Actually, there is a some
Please
disturbance,
Madam, in
keep
sir.
it loud.
Madam, in sugarcane,
Yeah.
we don't find any sucking pest, but we still find the poka boing disease.
Yeah, it must be in the ratoon crop. Is it in the ratoon
It is
crop
in
or the
ratoon crop
plant
only, madam.
crop?
Yeah,
Pardon?
yeah, madam, ratoon crop.
It is
Both
usually we
crop
have
we are
find
seeing.
with the ratoon crop. No, if you see by the adjacent nearby crop, you will find some insect because
Okay,
this poka boing disease transmission is still reported due to the some vectors.
madam, which virus it is?
Yellow mosaic virus, we say it is. I have written the name perhaps in my slide. Have you seen it? The
yes,
first
ma'am. It was written as
first few
fusarium.
slide. It is there, and we are working. We have taken a new project on this. So whether it is a phytoplasma origin, because one of our scientists has said it is the phytoplasma origin, another is saying that it is a viral origin. Doubt is there, work is going. So definitely, I cannot say what is what.
Madam, what is the actually management practices?
Yes.
Management practices, madam.
Yeah, please. For poka boing.
Yeah, madam, poka boing.
Yes, we do not have any this kind of recommendation till date, but it is better to do not take the seed from that field or remove the plant itself. The chemical control, etc. We do not have any recommendation on this yet today.
But madam, in what we did, urea and Boist,
You have to be a
spray,
little bit loud,
it
sir,
is
because I am not
recovered
able to hear.
in some of the crop. Madam, in some
Yes,
of the cases,
yes.
so what we did, we sprayed urea and carbendazim. So crop is recovered.
So how can you say it is poka boing affected crop? It could be some nutrient deficiency. It could be some problem with the soil. If the disease is viral oriented, it cannot be controlled through your urea or the fertilizer or the micronutrient management.
No,
It
madam,
could
even
be, its
carbendazim
symptoms
and
may
urea.
be your like yellow leaf disease, but it may not be the virus. It is possible.
Okay.
Here, without seeing this specimen or your plant sample, I cannot say definitely, but this is for sure. A virus cannot be cured through your urea or any other supplement. It could be symptoms could be there.
Okay.
Okay. Any other participants? If there are no queries more, Sigal sir, would you like to add something?
We thank
Hello, sir.
Dr. Shamila Roy.
Thank you very much, madam. On behalf of ICAR-NRIIPM and Manage Hyderabad, we sincerely
Hello, sir.
thank you.
Hello, sir.
Thank
Yes.
you.
Yes,
Thank
sir.
you, Doctor. Thank you for providing me this
Ask a
opportunity.
hello.
Yes,
Hello.
yes, Dr. Arthur sir, please.
Hello.
Yes, please, sir.
Yes, I would like to ask a question concerning the some topic concerning insects that attack sugarcane. Like
Please.
what biological control methods or chemical methods can you use to control these insects from attacking the same sugarcane crops? That's what I want to find out. Like
Actually, madam has delivered in detail about the biological measures and all the chemical measures in her presentation. Is it not?
Yeah, I'm asking because I didn't get it clearly. That is why I'm asking.
No, no, it is already time has been
So
us
it's
the
not that.
lecture notes. Listen, sir. Madam will be
Okay.
providing the lecture notes. The book will be released. You'll get to know the complete, detailed information regarding the pest management in sugarcane. That's okay? Is that okay?
All right. All right. If
Uh,
okay. Okay.
because already see, Madam has given a
Anyhow, uh, anyhow, Dr. Ravinder, we thank ma'am, right? And the other query will be taken, will take on the chat box or even mail to us. Then we will request ma'am to reply. Thank you very much. Now I request Dr. Ravinder, please introduce Dr. Doraemon.
okay.
Ma'am,
Dear
thank you very much.
participants, sir, can we proceed?
Sure.
Okay. Participants, now almost we are at the last session of our last technical session and last lecture of our training program. Rather, I would say it is IPM in oil seed crops, which is a very important crop of our day-to-day life. So this will be dealt by Dr. P. Dorai Murugan, principal scientist, agriculture entomologist, working in Indian Institute of Oil Seed Research, Hyderabad. Basically, his contribution is during last 17 years, he has involved in developing various eco-friendly pest management methods and IPM modules for cotton, pulses, oilseeds, and rose. He has identified and registered 6 leafhopper resistant castor genotypes with the Plant Germplasm Registration Committee (NBPGR). He is also associated in release of various castor resistant varieties of castors, which is resistant to leafhoppers. He has developed in vitro and in vivo methods for screening of leafhoppers and whiteflies, sucking pests with special reference, and bruchid resistance to mung bean and urd bean. He has also identified synthetic pheromones for attracting moths and major lepidopteran pests of castor and developed pheromone trap catch technology for Spodoptera littera. So presently he is dealing with development of water dispersible granular formulation of Bacillus thuringiensis for management of Spodoptera and development of semi-chemical based monitoring and management methods against major insect pests of castors. So he's also involved in screening and identification of dependable source of resistance, host plant resistance with special reference to castor, deciphering the associated mechanisms for resistance and mass production of Bacillus thuringiensis and Bavaria baciana formulation as oil-based suspension concentrates singly and in combination and field evaluation. He has various publication to his credit and he is honored with various various awards from different organizations from national and international reputes. And without wasting much time, we shall just get back to sir and we'll listen to sir and any further any queries we'll listen to him at the last. We'll have a discussion at the last. Please, sir. I will request the Emergan sir to please share his presentation, sir. Doraemon sir, please. Please unmute, sir. Please unmute yourself. Hello.
Am I audible? Am I
You're
audible now?
audible, sir. Yeah, you're audible. You're audible.
Yeah.
Please put into presentation mode, sir.
Yes. Is it visible now?
Yes, sir. Very much. Please go ahead, sir. Go ahead.
Thank you very much. Myself, Durai Murugan, working as scientist at Indian Institute of Oil Research in Hyderabad. In this lecture, I'm going to discuss about the pest management in oilseed crops. So coming to the status of oilseed crops in India, oilseed crops are second largest agriculture community after cereals. About 14% of the crop area is under these crops. In India, we have 9 annual oilseed crops, which includes 7 edible oilseed crops and 2 non-edible oilseed crops. Currently, it is grown in an area of 28.8 million hectares, with the production of about 36 million tons, with the average productivity of around 1,250 kg per hectare. Actually, compared to other crops, oilseed crops are relatively hardy crops. As you know, 72% of the oilseed crops grown under rainfed conditions. But still, it is susceptible to some of the insect pests and diseases. So, in this lecture, we are going to discuss about the diseases and the insect pests of these crops and how to manage these pests through integrated pest management. So, this is the production status of different oilseed crops in India and also sharing of different states in India. If you see here, the 3 crops, soybean, seed, mustard, and groundnut, these 3 crops produce almost contributes 90% of the oil, annual oil crops. So followed by castor, sesame, sunflower contributes at 10%, and this sunflower majorly is grown in a negligible area. But compared to the states, Madhya Pradesh, Rajasthan, Maharashtra, and Gujarat contributes almost 75% of the production in India. So, according to this status, I'm going to present you the 6 major annual oil crops and its problems in India. So, coming to the soybean, this crop contributes about 40% of our production in annual oil crops. So, if you see the insect pest damaging soybean, there are different groups of insect pests. The pest damaging the seedlings or stem, and leaf damaging insect pest, and some groups of sucking insect pests, and similarly group of pod damaging insect pests. So these are some of the major problems in soybean with respect to insect pests. So we will see one by one. So coming to the stem damaging insect pest, the important one is the stem fly. As you know, this adult looks like a horsefly, but it's a shiny in nature. It lays, especially it's a problem during the seeding stage, especially 15 to 20 days after sowing. Especially during July to August months, it is very severe. The adult lay eggs simply on the lower epidermis of the leaves. So the maggot, after hatching, it pour in the petiole, then it reaches the stem, and it leads to wilting and death of plants. So this is the symptom of damage caused by this pest, stem fly. It is one of the important pests, especially during the seeding stage. So coming to the next important pest which is damaging soybean stem is the griddle beetle. Actually, this pest starts from 25 days after sowing, but however, the damage you can see up to vegetation. That means up to 60 days after sowing you can see. But the female, this is the beetle, which makes the parallel. Here you can see the parallel grills are there in the stem or petiole of leaves. You can see, and it will make this griddle. That's why it is called a griddle beetle. Then it will lay eggs singly. So the portion which above the parts of the griddle will dry. So you can see the drying branches here and there in the field. Then the grab tunnel downwards and reaches the stem and reaches up to ground level and pupate inside only. So which leads to wilting of plants. Then in severe damage, all the plants will dry and die. So this is one of the important pests in many of the areas, soybean areas. So coming to the leaf damaging insect pest, you know, you have already exposed to several integrated pest management in several other crops. Similarly, this photo is a polypagous pest in different crops, including oilseed crops. So here it is damaged by defoliation. Initially, this larvae, you know, this moth lays eggs in egg masses. After a week, it will be in gregarious stages. After hatching, it feeds on wherever the eggs were laid in the plant. After that, it will spread to other fields and other plants of the fields. Then it leads to complete defoliation. So this is the nature of damage by the tobacco caterpillar in soybean. In some of the time, you can see the feeding of this larvae on pods also, and even birds or flower birds also. In some cases, it is a this. So this is another important pest, green semillipper. There are several species of green which is reported across India. There are several like Chrysotexis acuta, Dichrosia, Orichalcia, and Geosunia gemma. Based on the adults only you can differentiate, but even though you can differentiate through larvae to some extent. But one more brown semilipora also reported in India. So this all causes defoliation. Yeah. In the early stages, it leads to small growth, but when severe infestation, the total plants will be defoliated, leaving only the stem. So this is the important problem in soybean across India. The next important pest, the bigger caterpillar. As you know, this is a polypagous pest in several crops. Here also the same egg masses egg will be laid in masses, and up to 1 week it will be in the gregarious state. It feeds on the wherever the eggs were laid. You can easily notice the peppery leaves or brown yellow color leaves. You can easily locate from a distance. So in the severe infestation, it leads to complete defoliation, except stem and branches. But sometimes it damages the flowers and pods also in soybean. And another important pest is the leaf folder, but it is not a major problem. Occasionally, it causes damage in soybean. The larvae feed on leaves by folding them and webbing them. You can see here, this is the larvae. And the webbing of several leaves, especially at the apical end of the leaves, you can see. So this is the problem only in some of the locations, but it is not a major problem in many areas. So this is about the leaf damaging effect. Now we see the sucking pest. There are several sucking pests, especially they are important in terms of transmitting the diseases. The first important one is the whitefly, Pemisia tabacea. Yes. Now the nymphs and others suck the sap from the leaves and plants, but it leads to sickly appearance and stunted growth of plants. But it is important in terms of transmitting yellow mosaic virus diseases in soybean. Similarly, in some of the areas, one of the major problems, it sucks the sap from the stem and leaves and also in pods, which leads to yield reduction. Initially, the infested leaves are yellowing, curling, and wilting, and it leads to the stunting of plants and reduced pollen seed counts, and which leads to complete yellowing and drying of leaves. In some of the areas, thrip species like Thrips tabacea also cause damage. You can see the leaf with the whitish brown shining damage symptoms, which is followed by drying and dropping of leaves. Similarly, leaf hopper acid is also recorded in soybean in some of the areas, which leads to leaf yellowing and falling of leaves. So this is some of the important sucking pest infesting soybean. This is another important group of insect pest damaging pod in soybean and ground pod Helicobacter. As you know, it is a wide host. It is damaging almost all the important crops of pulses, oil seeds, and cotton and other vegetable crops and horticulture crops. Here in the case of soybean, it feeds on chlorophyll and young leaves initially and scutinize. Especially in the early stages, it defoliates the plant. It feeds on the leaves. But in later stages, it also feeds on the flowers and one more pod borer, Ticora also reported in some of the area, which is also causing similar damage to the pod. So this is all about the insect pest. Coming to the important diseases of soybean, this is the stem and root rot and charcoal rot. These are very important root rot diseases, especially which damage the collar region. So in the stem rot, sclerotium stem rot, the patches of plants with yellow dripping leaves you can see, and especially which is higher during high humidity area. It is soil-borne pathogen and it has wide host length. It is infesting several other oilseed crops also, which leads to complete wilting and drying of plants. Similarly, charcoal rot caused by macrophobina also, which leads to discoloration at the collar region of seedlings, especially between root and stem bases, which leads to wilting of plants. And you can easily see the small block sclerotia below the bark. So it is the seed and soil-borne pathogen, especially it is severe during and dry weather.
Sir, Dorai Murugan, sir.
Yes, sir.
Sir, your voice is not clear, sir. I think there is some network issue.
Sir, please rejoin.
Rejoin? Yeah. Sir, am I audible now? Is it clear?
Now it is clear, but in between, sir, during your lecture, the same, your voice will be somewhat disturbed, sir.
Now is it okay? Now it is
No,
okay.
as of now, it's okay, sir. As of now, it's okay. Please share your screen, sir.
Yeah. Now is it audible?
Yes, sir. Yes, sir.
Yeah. We will continue. Sorry for this inconvenience.
No, please, sir. Please carry.
So this is some of the important diseases of soybean. Coming to the next important disease is the rust. Actually, initially you can see the light brown pustules on the leaves, and which turns to reddish brown later. So which leads to complete yellowing and premature defoliation of the soybean leaves. So especially it is favored by 18 to 23 degrees Celsius with a relative humidity of 80%. This is one of the major diseases in soybean. The another important one is the frog eye leaf spot. Especially this is a Sarcospora species, which leads to, you can see here, ashy gray or olive gray in center with a dark reddish brown border. This is one of the important diseases, which in severe cases it coalesces together and it spreads to branches, stems and pods also. So it leads to complete defoliation of the leaves. So the another important disease is the bacterial pustule in some of the areas, especially this yellowish green lesions. You can see here with a reddish brown center, raised pustules. Here similarly it leads to yellowing of leaves and premature drop off. So which leads to which is actually transmitted. Actually, it is the seed-borne pathogen, and especially also by infected crop debris. So this is one of the problem. The other important virus disease is the yellow mosaic virus. Especially there are different virus species of causing this one, Mumbai yellow mosaic virus and soybean mosaic virus, which leads to, you can see the yellow patches and the yellow mottling of leaves, and especially during low temperature it is more severe. Especially it is transmitted by whitefly species, famous here Tamasi. So coming to the inherited pest management, now we have seen all the insect pest and diseases damaging the soybean. Now coming to the cultural options, what are the cultural options available? After the cultivation of this soybean, cleaning of stubbles followed by deep summer ploughing destroys the pupae. Most of the soybean pest pupae and are hibernating in the soil, especially stem fly, griddle beetle, pod borer, and tobacco caterpillar. All the pupae will be in the soil. So the summer ploughing is one of the important operation to be carried out to remove the hibernating stages of the insect pest and also the spores of the pathogens to the hot sunlight. Similarly, crop rotation with the non-leguminous crops, especially non-leguminous crops, it's very, very important for leaf miner management. In soybean, intercropping with asafoetida or the millets like maize or sorghum or pearl millet at the ratio of 4:2 ratio reduces cholera diseases, and it also conserves natural enemies of the oilseed crops. Similarly, use site-specific optimum fertilizers, those of NPK, especially, and also sulfur at the recommended rate to your locality. Similarly, excessive application of nitrogen fertilizer leads to infestation of all the pests and diseases. So, especially in griddle beetle and cebulifer endemic areas, intercropping with millets like maize or sorghum should be avoided. And wherever leaf miner, you can use a tobacco caterpillar problem, you can use castor as trap crop. Wherever leaf miner problem, you can use groundnut as a trap crop. Similarly, for a pod borer like Helicoverpa mithra, you can use marigold as trap crop. And for griddle beetle, danger can be used as a trap crop. So these are some of the trap cropping for the management of insect pest in soybean. Similarly, selection of insect resistant and tolerant varieties suitable for the locality is very, very important. Similarly, avoid pre-monsoon sowing and use optimum seed rate. Don't use excess seed rate, optimum seed rate, and the recommended plant spacing to reduce the stem flap infestation. As we come to the mechanical and some physical and behavioral approaches, the monitoring is very, very important in soybean. Especially remove and destroy the stem fly or little bit damaged plant body, especially in the early stages of the crop, and also the leaf with the egg mosses or gregarious stages of larvae, of tobacco caterpillar and hairy caterpillars, and even pod borer larvae. This looks very big in size. And the OME infected plants, mosaic virus infected plants, you can remove in the early stages, and it is or under remove it. Similarly, the another important aspect of bird purchase. On the erection of bird purchase at the rate of 25 per hectare, it will attract the predatory birds for preying on defoliator larvae. This is a kharif crop, rainy season crop. This is one of the important practices, because during rainy season we can't enter and spray the field. So the birds will take care of during that period. So erection of bird purchase is very, very important here. Similarly, use of sex pheromone trap at the rate of 10 traps per hectare for the early monitoring of Sporophylla helicorpa, and also even leaf miner pheromone traps are also available that can be used for even monitoring. And you can increase it to 20 to 25 per hectare for mouse trapping also. And similarly, yellow sticky traps also you can use it for monitoring whiteflies and aphids. Then installation of light trap, especially during the early season, it will be used for the collection destruction of the leaf miner mouse. So this is some of the mechanical and physical and behavioral approaches for management of the pest. Coming to the botanical and biological control, for seed treatment, you can use any Trichoderma species, Trichoderma viridis or Horsianum, at the rate of 10 gram per kg seed. And you can go for soil application also with 2.5 kg of Trichoderma, pre-incubated with 250 kg of farmland medium. So that will take care of the stem and collar diseases. Similarly, release of Telenomus remus and Trichoderma clonis 4 times at the rate of 50,000 per hectare at weekly intervals will effectively manage this Sporophylla. Similarly, wherever possible in the early stages of the crop, wherever egg mosses are noticed, immediately you can go for neem seed kernel extract 5% spray. That will take care of the early stage larvae, and also it is effective on sucking pest also. Similarly, wherever Sporophylla and helicorpa problem, you can use the Sporophylla NPV or helicorpa NPV at the rate of 250 LE per hectare, especially coinciding with the early stage larvae. It will give effective control. Similarly, you can spray BT formulations 1 kg per hectare, and even Bavaria Bassiana formulations 1 kg per hectare against the lepidopteran pest. Similarly, in soybean, there are several very good predators and parasitides are there. So better conserve biocontrol agent through minimum use of broad spectrum insecticides. And in our institute, we have developed BTK suspension concentration formulation, which is having wide host range of managing Helicorpa, Sporophylla, and Semilupers. And this we have developed all the data for commercialization, and it will be shortly commercialized for, especially for soybean. In soybean, we have tested in 5 locations. It was found promising in all the locations against lepidopteran pest. And especially we have recently got a patent for combination microbial formulation, which includes BT and Metarhizium relay, and plus BT plus Bavaria relay, which is also effective against Helicorpa megira and also on Semilupers. So these are some of the options in future for the management of the effective management of the pest in soybean, especially in organic farming. So the last option is the chemical control. So this would be treated as the last option. And whenever the ETL of the crop is reached, then only we can go for a spraying of chemical insecticide. And these are the ETL developed for the soybean pest for griddle beetle 10% infestation at flowering stage. For green semiluper 2 larvae per meter roll length at flowering stage. Similarly, for tobacco caterpillar, up to 4 larvae per meter roll length at pre-flowering stage you can consider as the ETL level. Similarly, for pod borer, 3 larvae per meter roll length at pod development stage is the ETL level. For leaf folder, wherever it is the problem, 8 to 9 folder leaves are per meter roll length is the ETL level. So now several conventional insecticides are banned or about to be banned. So you can always use the approved insecticide registered for the crop and for the particular pest. There are several insecticides, several number insecticides have been registered for this pest. This soybean crop, you can see here, including biopesticide BTK, that is having label claim for Sporophylla, Helicorpa, and a hairy caterpillar, Semilupper, and leaf miner, all the pests. Similarly, the newer insects like Spinodrum, it is having label claim for tobacco caterpillar. So based on the recommendations of CABRC, you can use it. Similarly, flubendiamide, there are different formulations for management of lepidopteran pest. Similarly, chlorantraniliprole, imamectin benzoate, also effective on several lepidopteran pests, including this chlorantraniliprole a very effective one, stem fly also, and brittle beetle also. This also it can be used in the IPM practices as the last option whenever it is a rigid the ET level or it is a endemic areas. So this can be updated every year, and the last it was updated during November 2021. So this is the latest registered insecticide for soybean. Also, coming to the registered fungicides, these are some of the newer fungicides registered for the management of soybean diseases, especially these flusa, pyroxide, hexaconazole, and there are several other single and combination fungicides are available for effective management of the diseases which we have seen now. So, as per the recommended doses, you can go for management of the diseases, and which includes some of the seed treatment chemicals also. Especially you can use this combination, including insecticide, combination fungicide and insecticide available for management of almost all the diseases, which is recommended at the rate of 10 ml per kg seed. Similarly, you can use the other recommended seed treatment chemicals or foliar spray the fungicides for effective management of the diseases in soybean, which is having a label claim for this particular crop. So this is all about the soybean. Coming to the next important oilseed crop is the rapeseed mustard. Actually, this contributes almost to 25% of the production contribution in India. But if you see, only mustard rapeseed is the major problem in almost all the rapeseed mustard growing areas. But however, the painted bug, it is also one of the major pests. Another important one is the mustard softly. It is not a regular problem, but occasionally it is reported in some of the areas. These are some of the minor insects like leaf miner, diamond back moth, leaf pepper, and cabbage head borer, and Bihari carry caterpillar. And these many of them are polypagous pest, and it also damages mustard when suitable weather conditions occurs. So the come to the important discipline that the insect pest is the mustard aphid. As you know, this you can see here the damage symptoms of the aphid, and the nymphs and adults sucks the sap from the leaves, stems, inflorescence, and developing parts, which leads to curling of leaves initially, and when followed by flowers fail. You can see the flowers damaged by the due to sucking nature of the aphid, which flowers fail to form pods, and the developments do not produce the healthy seeds, which is abundant during December to March month, which drastically reduces the yield and also oil content. So this is another important insect pest, painted bug. Actually, it occurs in two seasons, during seedling stage also and during harvesting stage also. Yeah, the nymphs and adults suck the sap from the leaves and also from developing parts. So which leads to the whitening of leaves, then it gradually wilt and dry off. So this pest you can easily see on the threshing floors also whenever after harvest. Normally it occurs especially during seedling stage October, November, then during the harvesting stage, especially during March to April. So this is one of the important pest problem in mustard. The another important and peculiar pest is the mustard softfly. It belongs to Hymenoptera. You can see the peculiar larvae feeding on the leaves by making irregular holes, which is sporadic in nature, especially during September to November months, and decline thereafter. Especially during Kharif season, it feeds on dry leaves. After that, it is shifted to the mustard and rapeseed crops. You can see this yellowish green or dark green larvae with stripes longitudinally. This is one of the peculiar Hymenopteran insect pest in mustard. The another polypagous pest, Bihari caterpillar. As I told in other crops, it is a cause complete defoliation. Similarly, pea leaf miner also cause damage on rapeseed mustard because both the crops occurs during Rabi season, which also cause leaf mining and zigzag mines due to the feeding. And because of heavy infestation, the leaves become yellowy, and it leads to falling of leaves. It leads to yield reduction. Similarly, diamond back moth. In some of the years, it is a major problem. You can see the larva mine the epidermal surface of leaves, and it produces white patches and feeding holes. The other is important is the leaf vapor that cause damage on leaves, buds, and pods by making webs and feeding on the leaves. So these are some of the polypagous pest infesting mustard. So coming to the important diseases of mustard, the first important one is the white rust. Actually, you can see the raised pustules, blisters you can see on the leaves, and it can be also seen on petiole and floral parts, which leads to deformity of floral parts. It becomes completely sterile, and especially late soon crop causes more damage. Similarly, this is the sclerosion of stem rot. You can see the rotten plant parts characterized by the presence of cottony mycelial growth of fungus on the stems and also on pods. So, which leads to the affected plant shows stunting and premature ripening and shredding of stem, wilting and drying. So, this is one of the important diseases. So, similarly, alternaria blight. This is initially starts with the leaves, then it spreads to pods also. You can see here the completely black and black colored pods. This seed sides will be reduced and discolored, and the oil content will be drastically reduced that the seeds won't germinate properly. Another important is the powdery mildew. Especially during warmer and drier drugs, it is a major problem, especially occurs during the reproductive stage during February to March month. And initially white patches on leaves, you can see on leaves, stem and pods. Under favorable conditions, especially a high temperature period, infection will be severe, covers the entire leaf, stem, and the sleeve are also affected by the diseases, powder mildew diseases. Similarly, the another important is the downy mildew. The susceptible stage is the flowering stage. Initially, it occurs on all the aerial parts. Usually, you can see on leaves and inflorescence. You can see the yellow irregular spots appear on the upper surface of the leaves. Similarly, you can see the white cushioning growth on the under surface of the leaves. If you see the affected inflorescence, it is small formed. It is small formed with white powder growth. You can see on this. So no pods are produced from this sterilized inflorescence. So the plants also looks stunted growth. So coming to the IPM in rapeseed mustard, there are several cultural options available, especially in rapeseed crop rotation, especially based on the location, area, suitable cereal crops can be used and avoid monoculture of mustard, which always causes sclerosis of temporal diseases. Similarly, grow early maturing varieties of Indian mustard as they are tolerant to mustard aphid and more yield potential than the rapeseed mustard, than the rapeseed. So similarly, early sowing before 15th October avoids many pest and diseases. Similarly, as I told in other crops, use of recommended fertilizer and well decomposed farm manure. This is one of the problem. If you want to properly use the well decomposed one, the termite problem will be more. So similarly, you have to destroy the plant debris in and around fields to reduce the termite infestation. Similarly, clean cultivation by weeding and destroying of debris in and around field reduces painted bug and softfly infestation. And another irrigation management is very, very important in mustard, especially first irrigation and 3 to 4 weeks after sowing is very important wherever painted bug is a problem. Similarly, timely irrigation helps in killing mustard softfly larvae through drowning. And another important is harvest at appropriate time, especially during 75% of the pods turned golden yellow color should be harvested, and it should not be properly threshed as early as possible, because keeping in the threshing floor will cause losses due to painted bug infestation. So coming to the mechanical loss, I told the monitoring of the crop, especially during December, January, is very, very important because this is a crucial stage. So during this period, you can see a lot of aphid infestation. So whenever the initial infestation, you can remove the twigs at 10 days interval to prevent the multiplication of aphid and spreading to other plants and crops, crop fields. So similarly, painted bugs normally aggregates on leaves and stem and even inflorescence that can be jerked into the carcinogenic water and can be killed. Similarly, leaf miner or you can easily collect and in the early stage and kill and prepare the maggots and prepare of this insect pest with the carcinogenic water. Similarly, this hairy caterpillars, leaf vapor, early stage, they are in a gregarious form that can be easily collected and can be managed by removing them from the crop field. So they come into the botanical and biological control. Soil application of trichoderma based product, it may be trichoderma horseanum or trichoderma viride at the rate of 2.5 kg per hectare, pre-incubated with the 50 kg farmyard menu, will reduce the soil-borne pathogen inoculum. Similarly, seed treatment with the trichoderma based product at the rate of 10 gram per kg, effective on soil-borne diseases. Similarly, neem oil 2% or NSK for 5% can be used to reduce the mustard aphid infestation. Even verticillium lichens, especially during 80% relative humidity time, you can use it, which effectively control aphid population up to which you control the aphid population. Similarly, release of some of the predators like Coccinella septum at the rate of 5,000 beetles per hectare or Chrysoperla will effectively check the aphid population. Similarly, there are several natural enemies are occurring in mustard, like ladybird beetles and cirripedes and lacewings. You have to conserve it. Similarly, pollinators are also very, very important in mustard. So use botanical and biological control agents to conserve these predators and pollinators. So coming to the chemical control, apply chemical control whenever the pest population reaches the ETA level. So especially spray in the evening hours to avoid toxicity to the insect pollinators. So for aphid, at least 10% of the plants with the shoot having 20 to 26 aphid in the 10 centimeters shoot only, you can go for any other management options for management of the aphid. Similarly, based on ACRP, seed treatment with the imidacloprid 5 gram per kg effective in controlling the painted bug up to 35 days after sowing. But these are the two insecticide which is having a label claim for the mustard crop. Imidacloprid, which is used for seed treatment, 7 gram per kg seed for effective management of softfly and painted bug. Similarly, thiamethoxam, it can be used as a spray, foliar spray for management of aphid. This is the use of registered fungicides for the management of diseases, especially the liver insects are approaching. It is registered for alternaria blight management. Whatever the recommended dose you can use for the management of the diseases. Similarly, there are several metallexyl fungicides are available for seed treatment also, and foliar spray also for management of white rust, alternaria blight, and also for down immunity diseases. These are the some of the approved fungicides. So these are this all about rapeseed mustard. Now we are going to the third important oilseed crop, groundnut. Here also we have different group of insect pests which is damaging the crop like above ground pot and below ground pot. And there are important defoliators and sucking pests which is causing damaging ground in the above ground pot, as many root and pod feeders, and the storage pests, some of storage pests which cause damage on the pots, that means below stored pots also, below ground pots. So coming to the defoliators, this leaf miner. Actually, here this leaf miner is a moth. It's a diligent moth. It is not a dipteran leaf miner. It is also called leaf webber. Actually, this leaf webber lays eggs on the tender leaves. You can see the tender leaves. Then first initially it starts the web together and feed remaining within the folds. In case of severe attack, it will cause burned-like appearance. You can see here, like a severely infested groundnut plant, and it peepers inside the leaves itself. This is one of the important problems in under input, especially in South India, especially in Andhra. It is one of the important problems in groundnut. So similarly, this product this we have seen in many crops. It is a polypagous pest. It lays eggs on egg masses, and initially it feeds on gregarious status, later spreads to the entire plant and damages the all the plant parts except the stem. So it peepers in the soil. The another important polypagous pest is the crop borer, especially in Helicoverpa. In case of groundnut, it feeds on the foliage, especially it prefers the flowers and buds. When the tender buds are eaten, symmetrical folds you can see on both the side, and you can see the unfolding. After unfolding leaves, you can see both the sides symmetrical folds. You can see. So this is the peculiar damage of the Helicoverpa in case of groundnut. This is the moth, especially this is the green tinge is the male moth, and the brown tinge is the female moth, which lay eggs normally singly in 5 to 10 eggs will be laid on the tender parts of the leaves, part of the plant, and peepers in the soil. So this is one of the important pests in groundnut. The other important pest is the red hairy caterpillar, though it is not a major problem in the recent years, but it is one of the potential pests in groundnut, especially during the sowing during sowing period. June, July month, the young larva feeds on the seedling, so which leads to re-sowing of the crop in some of the years. But in the later stage, it completely defoliate and cause damage to leaves, flowers, and growing points. The another important polypagous Bihar hairy caterpillar, similar to other crops, it also damages the groundnut crop by defoliation. Coming to the sucking pest, aphid. Aphid is important, especially in terms of vector. It is a vector of peanut strep viruses and also rosetti viruses in groundnut. In addition, it makes success from the tender parts, especially leaves, leaf buds, flowers, which leads to, you can see the stunted growth of the plants. And excretion of heavy honeydew leads to sooty mold development, which leads to block coating on the plants, which affects the photosynthesis, and ultimately it will affect the yield. So there are several species of thrips species like Phytothrips, thrips pomae, and Calithrips are recorded on groundnut. It also involved in transmitting some of the diseases like bud necrosis diseases, and it leads to. And you can see, you can usually differentiate, especially in the tender leaves, the yellowish green patches you can see on the upper surface, and you can see the brown necrotic leaches, and on the back side you can see the silvery seen on the lower surface. Those severe infestation cause stunted growth. So this is one of the important pests and also a vector of some of the viral diseases. The another important is the jacid or leafhopper. Sometimes the damage will be confused with the micronutrient deficiency. You can see the V-shaped here on the tip. You can see the yellowing, the V-shaped yellowing. This is the peculiar damage caused by the hopper band due to its hopper band injury or toxins produced by the leafhopper. So this is all about sucking pest. Coming to the next group, major group is the root and pod feeder. This is very, very important because it completely causes direct losses to the plants and leads to yield loss. So this white grape, as you told, several IPM crops you have seen. It is a major, one of the major pests in several crops. Even recently, sugarcane also we have seen the pest. Similarly, it attacks groundnut also, especially sandy soil. It is a major problem. This is the 1 year life period, this life cycle, especially this adult beetles emerges during May month and lay eggs in the soil. Initially, it feeds on grasses. Later, when the crop was sown during June-July month, it caused damage on the groundnut. The grapes feed on fine rootlets, which leads to wilting of plants and drying in patches. And you can see the grape moves straight row. So you can see the damage in the straight in the row wise. You can see the damage with the wilting of plants. If you remove the plants, you can see the grape inside the soil. So this is damage about root and pod feeders under this white grape. And a similar termite. This is one of the major problem which leads to wilting of plants, especially under where the plants plants also here damages the pods and damage the seeds. That is very, very important in case of groundnut. The another important one is the storage pest, especially though it is a storage pest, but the infestation starts from the field and it is carried over to the store. So here, this is entirely different from the pulse beetles, which is infesting like other pulse crops. But here it pupates outside the pod or seed or the gunny bags. So this is one of the important insect pests which damages the both pod and seeds of the groundnut. Actually, it starts from the field and cause heavy damage under storage conditions. So coming to the important disease, though there are several diseases of there, especially this leaf spot diseases, stem rot and necrosis are very, very important. So one by one we'll see that. Coming to the Tika disease, in this there are two, early leaf spot and the late leaf spot. Actually, this early leaf, this is the symptom of early leaf spot diseases. It appears especially during 1 month after sowing. You can see the small the spots appear on the leaflets initially, and later it yellow and they turn brown to block on upper surface of the leaves. These spots are circular to irregular shape, by especially characterized by yellow hollow of various width. In severe conditions, it appears on the even in petiole stems and stems also, and which leads to premature senescence and shedding of leaflets. This is one of the important diseases. Similarly, late leaf spot, especially its infection starts after 55 days in Kharif season and after 40 days in Rabi season, and it is especially epidemic during maturity stage. And you can see this here block and circular spots you can see on the lower surface of the leaflets, and a distinctly round spot and yellow colors you can see on the mature spots like this one. So spots are almost black on both the surface. In extreme case, like a early leaf spot, this premature senescence occurs and shedding of all the leaflets occurs. Similarly, it's a prolonged high relative humidity for 3 days and low temperature of 20 degrees Celsius and a heavy application of nitrogen phosphorus fertilizers and a deficiency of magnesium in soil favors the diseases. So the rust is another important diseases in groundnut. You can see the orange or red or brown pustules you can see on the lower surface of leaves initially. Later, these pustules are also seen on the upper surface of leaves also, and on the other aerial parts also. The leaf curl and drop off, resulting in complete defoliation. Similarly, you can see the stem rot also. especially during 70 days after sowing, you can see. Initially develop the brown lesion, you can see here around the main stem at the soil level, and then the lesion become covered with white radiating mycelium, you can see here. And after post, this it leads to wilting symptoms characterized by yellowing and browning of foliage, then followed by wilting and drying of plants. Similarly, as I told, this peanut buggerosis disease is also very, very important, which leads to stunted growth with shortened internodes and auxiliary shoots. They completely affect the yield. You can see this is also infects several weed species like Porcinium, Erigan, and Camellia, Tagetes, and Trifolium weed species. It is transmitted by thrips species, and especially the population occurs during both Kharif and Rabi seasons. So it is even spring and summer season also. It causes damage in almost all the seasons, both the seasons. So coming to the IPM in groundnut, this cultural products like use of trap crops, like wherever leaf miner and leaf hopper problem, you can use a cowpea or soybean as a trap crop for in the groundnut for managing the pest. Wherever spodomtra problem, you can use castor as a border crop. Similarly, growing of millets like tall crops like pearl millet, sorghum, or maize, reduce the incidence of thrips. It is a vector of peanut stem necrosis disease, as we have seen. Similarly, this vegetative trapping is very, very important, especially wherever red hairy caterpillar is an endemic problem. You can use tweaks of Jatropha or Calotropis. You can use so that it is attracted to the even in the field or. This is can be used for managing the red hairy caterpillar. Similarly, wherever this white grub is problem, especially May month, you can use the neem branches for trapping of the white grub. Then it can be killed in putting the kerosene water. Similarly, intercropping of castor or red grub in the ratio of 1:11 also recommended in groundnut crop for management of the diseases. Similarly, for the management of diseases, you can use millets, pearl millet, sorghum with the groundnut 1:3 ratio. Introduce the early use spot and rust diseases. Crop rotation is very, very important, especially with the suitable cereals. Similarly, intercropping of red gram and bajra or red gram and castor with the groundnut, or alternate cropping with the millets, very, very important to reduce the stem rot diseases, especially in the endemic areas. At least once in 3 years, it should be crop rotation should be followed with the other millet crops. Similarly, removal of voluntary crop plants and self-grown groundnut plants and other weed plants having alternate hosts should be removed to reduce the primary source of infection. As with soil application of castor cake or neem cake at the rate of 200 kg per hectare also recommended for management of the diseases. As I told in other crops, collection and destruction of the egg mosses, early early stages, and disease initially disease infected plants should be removed and burned. As well as bird purchase, especially in groundnut 25 bird purchase, you can install at the to prevent the to manage the leptopteran pest and other pests. Similarly, you can use polyethylene gunny bags instead of jute bags that will give control against storage pest like groundnut bruchid. Also, coming to the behavioral and botanical options, you can use for Podomtra helicorpa, pheromone traps. You can use 10 traps per hectare for monitoring, or 25 traps you can use for moss detection, especially for leaf miner. Aprorimma modicilla, you can use 25 traps, pheromone traps for moss trapping of the leaf miner moths. And you can use light trap, especially for during monsoon season, which effectively controls the red hairy caterpillar and also white grub. Similarly, white grub aggregation pheromone is there, anisole is there. Whenever this continuously for 3 days, you can keep it after the first monsoon. That will trap the adult beetles that can be killed in the used insecticide along with the pheromone dispenser. Similarly, spraying of NSK or any commercial neem formulation or neem oil 3 ml per liter, or azadirachtin, any neem formulation 3 ml per liter, or pungami oil 3 ml per liter, with some sticky agents like detergent powder 1 gram per liter, which is effective against defoliator and sucking pest. Similarly, seed treatment with the Trichoderma hircium 10 gram per kg seed effectively check the root rot population, root rot diseases, and even Pseudomonas fluorescens seed treatment with 10 gram per kg are reported to be effective against rust diseases in the groundnut. Release of Trichoderma culverins at the rate of 50,000 per hectare, 10 days interval also effective against Spodomtra litra. Similarly, release of Trichoderma remus, especially 50,000 per hectare, 10 days interval, which effectively check the Sporopter lectura. And wherever leaf miner and other defoliators problem, you can use the Bracon habiter. Release of adults at the rate of 5,000 per hectare, and 10 days interval, it will control the Leptopteran pest. Similarly, Sporoptera NPV, Helicorpa NPV, even Red Hairy Caterpillar, Nebraska NPV also you can use for management of the Leptopteran pest, or you can use BT also, 1 gram per liter for management of the this Leptopteran pest, especially coinciding with the early stages of the insect pest. Similarly, Metarhizium relay, which is very effective on Sporoptera litera, that can be used wherever high relative humidity, that can be used as a option for organic pest management. So this is the some of the ETL levels worked out for the groundnut pest, especially for leaf miner, up to 15 mines per plant also during 55 days after sowing. There will not be, then there is no need. Below that, there is no need for any spraying of chemical, and naturally it will be taken care and the yield loss will not occur. Similarly, for defoliation, 10% foliage damage, you can consider as ETL. For Sporoptera, 20 to 25% defoliation at 40 days after sowing considered as ETL level. Especially for the sucking pest, it is very, very crucial because they are all vectors. So 5 to 10 aphids per terminal bud. You at seedling stage, you can go for any other management options, including chemicals. Similarly, for acids, 15 to 20 acids per plant. For thrips, 5 adults per terminal buds. For white grape, it's very, very per meter squared. Even 1 grape also considered as ETL level for taking pest management options. So this is the under ACRP groundnut during 2020. This all the technologies recommended for management of sucking pests like aphid, acid thrips, seed treatment with the imidacloprid, thiamethoxam, 2 ml per kg. It is recommended. Similarly, foliar spray of these insecticides is also recommended at the rate of 0.3 ml per liter of water. For white grape management, seed treatment with the imidacloprid, 2 ml per kg of seeds recommended. For management of leaf miner, defoliators, and hairy caterpillar, flubendiamide or chlorantraniliprole 0.2 to 0.3 ml per liter is recommended. Similarly, you can use the growth regulators like Nauduron or spinosad, say for insecticide 0.3 ml per liter also can be used for management of the diseases for the insect pest. Similarly, for storage pest, especially wherever you are storing with the common good bags, you can go with a infecting the bags with the deltamethrin 0.5 ml per liter, which protect the groundnut, which especially for seed purpose stored for up to 6 months. So this is the technology development for ACRP groundnut. But if you see the registered approved insecticides for groundnut under CABRC, some of many newer insecticides are registered like flubendiamide for Sporoptera litera, and even for sucking pest, thiamethoxam has been registered, and even some of the areas rat is a major problem. So bromondailan also registered for management of the rat problems in groundnut, and this is having the labeled claim for groundnut crops. Similarly, methoxyphenoside also recommended for Leptopteran pest like leaf eating caterpillar like Sporoptera and leaf miner. These are the registered approved insecticides. Similarly, several fungicides are registered for groundnut crop. Here's especially this chlorothalonil for rust and tick management, difenoconazole, hexaconazole, they're all registered for the especially leaf spot and rust diseases. Similarly, propiconazole, it is for leaf spot and rust diseases, it is recommended. Even several sulfur formulations. Though I have mentioned here one, there are several sulfur formulations available for management of tick leaf spot. Similarly, some of the seed treatment chemical are also available for the seed treatment like mango stem. This is a conventional one that can be used for seed treatment also. Similarly, tebuconazole seed treatment and foliar spray are also available for management of the diseases in groundnut. So interestingly, there are 3 biopesticides also having labeled claim for groundnut disease management, especially Pseudomonas fluorescens, bettable powder formulation for leaf spot diseases, late leaf spot diseases, through seed treatment, 10 gram per kg seed. Similarly, soil application, 1 kg per hectare also recommended. And again, another strain also registered for management of leaf spot diseases. And another Trichoderma strain, Trichoderma vredi, also registered for stealing wilt diseases through seed treatment and also through soil application. So these are some of the biopesticides for management of diseases in groundnut. So this is all about the 3 major oilseed growing crops. Now we will see about the minor oilseed crops, which includes The minor oilseed crops include there are some sesamum, which is also called as sesamum gingerly. But if you see the insect pest, only leaf webber and capsule borer is the major problem. But otherwise, godfly in some of the areas, it is a problem causing up to 15% yield loss. And the leaf hopper is a problem, especially in terms of vector. It is a vector of phyllotic diseases. And the sesamum caterpillar or Sphinx caterpillar, it is a problem in especially in UP areas, and it is a sporadic nature. Suddenly, it will cause severe damage in case of sesamum. Similarly, Bihar hairy caterpillar also reported on sesamum. So we will see one by one the damage symptoms. This is the leaf webber. Leaf webber and capsule borer, it is called a leaf roller and a capsule borer, and also shortly called as anticastra. This is one of the major problem in sesamum area, and because it causes damaging throughout the season from seedlings to harvest. So initially the larva feed on tender leaves by remaining inside the leaf web. Initially, this is the damage. You can see the larva here webbing of the leaves. And flowering stage, it damages the flower. You can see the feeding nature of this larvae on their sesamum flower. And during capsule formation stage, the larvae bore into the capsule. You can see here. When it is severe case, it completely damages the crop. This is a complete yield loss, even up to 100% yield loss reported in some of the areas. This is the adult moth, which lays about 8 to 10 eggs singly on the lower surface of the leaves. This is the larvae, spotted larvae you can see. And this is the pupa you can see inside the leaf webs. Webs you can see easily. This is the important pest in as some leaf webber and capsule borer. The another important one is the gallfly, Aspendelia sesame. Even some of the areas, butterfly, Desdura sesame also reported, but the damage is both are depleted pest. This is the, you can see the fly, which lays eggs on the flower buds, especially maggot feed in the floral bud, then maggots feeds inside the floral bud. Because of this feeding nature of the maggot, it leads to complete gall-like structure, which do not develop into flowers and capsules. So the affected buds wither and drop. So this is the healthy capsule, and you can see this is the damaged capsule by the gallfly. So this is some very important insect pest in sesamum. The another important one is a Sphinx caterpillar or hawk moth, or it is also called a deadhead moth, because you can see the deadhead like this the structures on the thorax of the moth, and this is the cause complete defoliate. So voracious feeder, it defoliates the plant completely. It prepares on the soil. This is one of the important problems specific to this sesame. This is this insect pest. And coming to the another important polypus, Mr. Bihar hairy caterpillar. As I told you, the moth lays eggs on mosses. So the gregarious stages, it feeds completely all in one place, and after that, you will spread to the other parts. So the mature insects occur from vegetative to maturity stage, and it causes defoliation. Another important sucking pest is the leaf hopper. This is the Voracious albicicus. It is also called white leaf hopper. This is the Nimsa leaf hopper. It also sucks the sap from the tender parts. You can see here the damage of the leaf hopper. The bottom leaves, you can see the yellowing and the cupping, curling of the leaf edges and drying of leaves. And in the later stage, it transmits the phyllotic diseases, phytoplasma diseases. So it is important, especially in terms of vector. So coming to the diseases, there are several important diseases are there in sesamum, especially stem and root rot. Macrophobia root rot is very, very important, especially it appears on the root and stem part. You can see the yellowing and drooping of leaves initially in the plants. On the ground level, you can see the stem become blocked and so rotting symptoms here. You can see if the plants are uprooted, you can see the block colored roots having the sclerosia. It's like a charcoal sprinkled on the root. So that's why it's called charcoal rot also sometimes. So this is important diseases in several sesamum growing areas. And the another important disease is the wilt. Here it is the Fusarium oxysporum sesame. Initially, it causes the yellowing of leaves, droop down of plants, and which leads to complete wilting and drying of plants. If you see the root, you can see the vascular discoloration and rotting, and you can see the pinhead sized sporia over the dried stem. That is the to differentiate it from the other diseases. This is the wilt diseases. Similarly, field downing, it is one of the very, very important diseases in sesamum. because all floral pods transform to green leafy structure. It looks like a witch's broom, which do not bear any capsules. Even the capsules are formed which do not yield quality seeds. As I told, it is transmitted by Leafhopper voracious albicenter. In some of the areas, some other species like Ecusimonus also transmitting the diseases. The another important disease is the powdery mildew. It is emerging as a major problem in several areas. And you can see the small cottony spot appears on the infected leaves, which gradually spreads on the entire lamina, which is fully covered with the white powdery moss, which leads to complete defoliation of the plant before maturity without any producing flowers and capsules. So this is another important diseases. So there are several leaf spot diseases reported on some, especially Sarcospora leaf spot, which initially occurs as a small angular brown spots with the gray centers and dark margin. And when severe conditions, it spreads to leaf petiole, stem and capsules also, which leads to defoliation and loss of the yield. Similarly, ordinary leaf spot also recorded. You can see the brown circular to irregular shaped spot when this concentric rings. Especially the spot appear when the crop is nearly 1 month old, which leads to defoliation of the leaves. Similarly, there are blight diseases like Phytophthora blight and Xanthomonas bacterial blight also recorded. Especially in both the cases, the water-soaked spots appears on the leaves and stem, and which leads to black-colored leaves and appears stem near soil level. Then they spreads to other branches, leads to death of plants. Similarly, in the bacterial blight also, the irregular spots you can see on the leaves. Later, it increases in number. You can see, and then it completely turned brown. Under favorable conditions where the severe infection, the leaves will defoliated from the plants. So this is some of the important diseases. So coming to the IPM in sesame, especially in sesame, crop rotation is very, very important. It reduces the pest and diseases. So at least once in a year, the sesame crop rotation should be followed, and sesame should not be cultivated in the same land year after year. Similarly, as I told you in other crops, deep summer plowing exposes several soil-borne pests and fungal propagules to high temperature of sun as well as to predator that will take care of the insect pests and disease-causing pathogens. Similarly, in Kharif season, early sowing immediately after monsoon and the use of early maturing sesame varieties will reduce several pest and diseases. Similarly, recommended for the region, use tolerant varieties and disease-free seeds. Similarly, intercropping with the mung bean, you can see intercropping with the mung bean or urud bean or cluster bean or groundnut, pigeon pea, pearl mill reduces the damage by insect pests and diseases compared to the sole crop. Similarly, sanitation, clean cultivation like a removal of Parthenium, Protozoa trifolium. These are all alternatives of Parthenium, the alternatives to Phytoplasma diseases and other diseases. This should be removed near the field and inside the field also. So similarly, as I told in other crops, collection destruction of egg mosses, early instars larvae, or bigger larvae like a spring scatter, which is easily visible to naked eye, and even leaf rolls and shoots birds of leaf webber or phyllodes infected plants in the early stages. If you remove and destroy, it will reduce the spread of the insect pests and diseases. Similarly, removing the galls and burning them are a good prophylactic measures to against the gallfly. So always light drop 1 drop per hectare for monitoring moss trapping of the moths. It will be operated during 6 to 10 p.m., especially coinciding with the early monsoon season. That will reduces the insect pest incidence in the later stages. The another important, especially in sesame, is the bird perch, because this predatory predatory birds are very effective in controlling the leaf webber and capsule. It prefers the caterpillar. So my installation of 50 bird perches per hectare completely checks the leaf webber population in sesame. Similarly, seed treatment with the Trichoderma viridi or Trichoderma horseanum or even Bacillus subtilis at the rate of 4 grams per kg against Phytophthora blight diseases and stem and root rot diseases, it is effective. And application of neem cake 200 kg per hectare effective against pest and diseases. As well, soil application of Pseudomonas or Trichoderma 2.5 kg mixed with 25 kg of well decomposed form manure 30 days after sowing, also effective against wilt and root rot diseases. Similarly, spraying of neem seed kernel extract or neem oil up to 5 ml per liter or commercial neem formulation 3 ml per liter effective against the insect pest. It also reduces the disease incidence. Similarly, spray of BTK, BT 1 gram per liter of water, we effectively checks the leaf weapon and hairy caterpillars. Similarly, wherever sesame seeds are also affected by some of storage pests like rice moth and red flower beetle, that can be managed with use of neem leaf powder 25 gram per kg seed, or neem seed kernel powder 5 gram per kg seed, or even sweet flag rhizome powder 10 gram. This is Acorus calamus, this is the scientific name, 10 gram per kg seed for the effective against the storage pests of sesame that you can control using botanical and biological options. So coming to the chemical control, application of pesticide only should be a last option, and it should be used only after crossing the ETL. If 10% damage or 1 to 2 larvae per plants are observed or 2 webbed leaves are observed per square meter, then you can go for pest management options. Similarly, for gall fly, 2 to 5 galls per plant or 10% damage considers ETL. But in the ACRP, there are several recommendations like seed treatment with imidacloprid 7.5 gram per kg seed or thiopenthoxam 5 gram per kg seed effective against the sucking pest. And similarly, spraying of a chlorantraniliprole or navluron or flubendiamide or spinosad, they are very effective against leaf webber and capsule borer. Similarly, for sucking pests like gold fly, bud fly, leaf hopper, spraying of imidacloprid or acetamiprid or thiopenthoxam or thiacloprid also, especially during but initiation stage, very effective for management of the sucking pest. Similarly, use of indoxacarb for management of Bihar hairy caterpillar and the Sphinx caterpillar also recommended. And seed treatment with metalaxate 6 gram per kg recommended for Phytophthora blight. But these are recommended based on ACRP sesame recommendations, but there is no label claim for sesame. But in case of sesame, no fungicides have label claim for some diseases, even for insect pest. 2 conventionally influenced, very old conventional insecticides like Winalphos and Acrydameton only having registered for sesame crop, but even this will be phased out in the coming years. So this is all about pest management using chemical control in sesame crop. So coming to the another important crop is the castor. This area is increasing, especially already you know Gujarat is the one of the major castor growing areas. Now it is popularized in Rajasthan, Haryana, also even in South India, Tamil Nadu, Karnataka, Telangana. Also it is getting popular because it is not affected. It is under rainfall conditions, very suitable crop, and there are several new hybrids are available, which is a very good yield potential. But you know, this castor already grown as a trap crop in many other crop ecosystem. But if you grow as a sole crop, you have to you can see several insect pests also. But it can be easily controlled with the least pest management options. There is no problem at all. So there are 3 groups of insect pests like defoliator, capsule borer, and sucking pest. So the first defoliator, the important one is the semilooper, castor semilooper. Mostly it prefers castor. Also, it is reported in pomegranate and rose crops. Initially, this is you can see the wax coated semilooper larvae. Initially, it nibbles the leaves, and the second it start damage the leaf by making holes. Late it start voracious feeder. It totally defoliate the plant. This is the damage by the semilooper in castor. This is the tobacco caterpillar. So as I told you, many crops, it highly prefers castor for egg laying and feeding. This is the gregarious stages larvae and damage leaves. Later stage is spreads to other plants and cause heavy defoliation. Some of the years it also feed on stems and capsules also. And there are very good naturalisms are occurring in castor, especially Appendelus parasitide for Sporoptelectra. It is effective parasitide. And the capsule borer is one of the major problem, especially late shoot crop. Wherever this August to shoot crop, it is one of the major problem. The larvae bores directly into the capsule, and you can see and web the complete spike and cause the damage. Sometimes it feeds on the shoot also. It bores and damage the shoot, but it is least preferred wherever capsule it prefers capsule only. It is causing direct damage to the spike capsules and feed the mature seeds. Similarly, leafhopper, it is one of the important sucking pest. In the recent years, it is becoming popular in both Kharif and Rabi seasons. because now this crop recommended for rabi seasons also under drip irrigation system. So it is becoming a major problem in castor crop. The nymphs and success sap from the plants, and the hopper bed symptoms are noticed, and the initial symptoms are yellow patches on the leaf margin, followed by leaf curling. Yellow color later changes to brown color, and it leads to complete drying and brittle on margins. Similarly, thrips, it is becoming a major problem in Gujarat. Nymphs and other feeds the terminal leaves and also on the floral parts, which leads to resulting distortion and characteristic wrinkling of leaves and drying of flowers without forming any capsules. This is one of the emerging problem in especially in Gujarat and also in South India. Wherever there is a prolonged dry spell, this is a problem. And similarly, whitefly, Triad rhodus racing, is a specific species to castor. It is also emerging as a problem in both Kharif and Rabi crops, especially in Rabi or spring summer rain is a major problem. The nymphs, and you can see here the nymphs and success sap from the undersurface of the leaves and causing yellowing and drying of the leaves. In severe infestation, because of honeydew secretion, it promotes block shooting mode, which interferes with the photosynthesis. So coming to the important diseases, there are three important diseases like fusarium wilt. Initially the yellowing leaves you can see, followed by wilting. It appears in patches and affects the all stages of crop growth. Similarly, alternaria blight, especially during seeding season, is very important. Light brown circular spots appear, which are irregular in shape and have concentrated rings surrounded by yellow hollow. It also damages stem, leaves, and inflorescence and capsules also. Another important disease in castor is the grey rot. Because of these diseases, the area declined, especially in South India, especially in Andhra and Telangana. The grey rot disease caused by Botulinia resini. It is previously called Botrytis resini. Especially, you can see the woolly growth of on capsules, which subsequently fall, rot and fall. Especially this disease, wherever wet conditions prevail with the high relative humidity of 90% and a cool temperature of 25 to 23 degrees Celsius during flowering and spike formation stage or mature stage, it is very, very important. If there is a cloudy weather for 3 to 4 days coinciding with this flowering or spike formation stage, that complete loss of this due to this disease. So coming to the IPM in castor, actually in castor, interestingly we have waxy type, and actually we used to call this bloom, waxy bloom. We have several varieties with bloom, without bloom. That means with vacciners, without vacciners. So based on that, there are several resistant, because this wax is directly linked to insects and diseases. If actually it is, there are several categories. If there is no wax at all on the plants, it is called a no bloom or zero bloom. If it is only on the stem, it is called a single bloom. If it is on the stem and lower part, it is called a double bloom. If it is in a stem lower and upper part, it is called triple bloom. So these zero and single bloom types are susceptible to leaf hopper and thrips. Here you can see the damage on zero bloom nature, but this is resistant. This triple bloom waxy genotypes are resistant to leaf hopper and thrips. But interestingly, this triple bloom are susceptible to whitefly. Similarly, with the compact spike, wherever the compact spike with the vaccinus, it is susceptible to capsule borer and grey rot. Wherever the loose type and non-spiny type of spikes, they are resistant to capsule borer and grey mold. So based on the endemic nature of the pest and diseases, you can select your cultivar with this background. So this is the, you can see the triple bloom castor without any spray. Here completely dried, but it's able to yield this crop with the bloom nature. This is the reaction to leaf hopper diseases. This is one of the popular hybrid DCH519 castor cultivar. Similarly, under cultural conditions, you can go for trap cropping for management of red hairy caterpillar. Sowing of cucumber all along the field for attract a migrating red hairy caterpillar. This can be used in other crops also like groundnut. So wherever it is possible, that initially this cucumber attracted caterpillars can be put it in carbonated water and it can be killed. Similarly, in mechanical collection and destruction of the mosses, damaged leaves along with the gregarious larvae, and grey mold, grey rot infected spikes, and should be removed initially where occurs, and it prevents the spread of the insect pests and diseases. Similarly, bad purchase also recommended for a castor. It encourages the predatory bird for a management of all lepidopteran pests, including capsule borer. It also prefers capsule borer, so it can be used as an option for management of the capsule borer in castor. So, similarly, behavior control, you can use Lytra or management of red hairy caterpillar, or you can use Petromax lamb also, or nowadays solar operated Lytraps are available that cannot be also used for trapping the red hairy caterpillar wherever the endemic areas. Similarly, pheromone traps you can use for monitoring and mouse trapping of the moths. And there are seed treatment with Trichoderma harcianum 10 gram per kg seed or soil application of Trichoderma 2.5 kg mixed with the farmyard manure or spray of Trichoderma harcianum 5 gram per liter of water, especially spike development stage effective on grey mold diseases. Similarly, BT, several formulations we have developed in our institute which are effective on semilooper and early install RVOs, Propter. Similarly, NPV you can use for Propter and Numeria relay you can use for Propter management. And interestingly, in castor semilooper, we have very good parasitide. It is called microplatis parasitide. It is very unique and very potential parasitide. In some of the years, it gives 100% parasite. You can see the semilooper, which is the cocoon. This is the adult parasitide. Actually, it's the internal parasitide. After pupation, it will come and stick on the petiole or other leaf pods. So it cannot move further, and without food, it will die. So this is one of the important parasitide. If you see in the field 1 to parasitide per plant, no need for any spray of chemicals. As really egg parasitide Trichoderma also effective in semilooper management, but in case of castor, it is not economical. Similarly, even though castor is susceptible to insect pest, the one advantage is it can tolerate defoliation up to 25%. Even mechanically, if you remove 25% leaf also, there will not be any problem in terms of yield. So capsule borer damage up to 10% also you can use as a ETL level. So avoid using insecticide if the damage are below the ETL level. And through ACRB we have recommended full bendiamide, chlorant and chlorant spinosad for management of lepidopteran pests like defoliator, hairy caterpillar, capsule borer. For management of sucking pest, you can use astamiprid or clothianidine. That can be effective against sucking pests like leafhopper, thrips, and whitefly. As in castor industrial 1 BTK, it is our BTK, our product registered having label claim for hairy caterpillars and semilooper. And those there are some other insecticides are having label claim, but they are phased out or going to be phased out. So here that's why I'm not indicated. But none of the fungicides are registered for castor crop. Since it is a minor crop, no fungicides have been registered for this crop. So come to the last crop, sunflower. Actually, sunflower, there are several group of insect pests like sealing pest, sucking pest, defoliators, and pest on inflorescence. Coming to the sealing pest, this cutworm, agrotis is a major problem, especially in the potato growing areas in northern India. The larva cut the sealings at below ground level, which leads to death of affected plants, which leads to the problem in plant stand, which leads to resowing of the crop. Especially it is a problem in potato growing areas. In other areas, it is not at all a problem. Sucking pest, especially this leafhopper, it is emerging as one of the major problem in both Kharif and Rabi crops. Especially it's a problem in Rabi crop. Both limbs, you can see the limbs and adult suck the sap from the plants. Initially, yellow patches you can see on the leaf margin, and later you can see the drying of leaves and brittling on the margins. So you can see the hopper band symptoms. The heavy infestation leads to loss of vitality and poor seed development. About 30% yield loss has been reported due to this pest during Rabi season. Similarly, thrips, especially this is important in sunflower because it is a vector of sunflower necrosis disease. There are several thrip species infesting this sunflower crop and also damages by sucking the saps from the tender parts of the plants, also as a vector. And coming to the defoliator pest, this is the green semilooper Thysanopodicea, and it's one of the important defoliator. Initially, it feeds on chlorophyll, later it completely defoliate the plants, leaving only midribs. And similarly, tobacco caterpillar, like other crops, it damages the sunflower crop also. And another important pest in sunflower is the Helicoverpa, which is damaging on the head. That means capitulum. The larvae bore in the head and feeds on the developing seeds. In the larvae feeds inside the early stage of growth and move towards the capitulum. And initially, this is called starboard stage. This is very crucial stage. In that stage, it will be closed, so the damage won't be visible. If it is not noticed, then complete loss occurs. So this starboard stage is very, very important. After opening of the flower, normally natural image will take care, but this starboard stage is very, very crucial. It should be checked and it should be managed properly. And similarly, this parakeet is one of the major problem in sunflower wherever it is grown as an isolated field. Actually, if it is more than 25 hectare area in any of the place, the damage is only less than 5%. But if it is the isolated field of 1 hectare or 2 hectare, the damage may be more than 90%. The bad damage start from the milky stage to till harvest, it will cause damage. This is one of the important problem in especially in sunflower. So this there are some other diseases also like other crops, the necrosis diseases, especially this is transmitted by tobacco streak virus. This is very, very important. Transmitted by Thrips species. It leads to necrosis and twisting of leaves, which will not give yield. Similarly, leaf blight also one of the problem, which leads to blightening of leaves. Powdery mildew, white spots on the upper surface will completely cover the plants, which leads to complete defoliation and without any yield or formation of head. So these are some of the important diseases in sunflower, and these are some of the insect diseases important in some of the areas like downy mildew. It's a major problem in Maharashtra. Maharashtra and some of the areas of Karnataka and Andhra Pradesh, and the head rot is a problem in Punjab. And the stem borer, it is a typical problem in Maharashtra, Latur region. There, the collar region enter the grub enter and damages, and the infested plant lodge during grain formation stage. So these are some of the specific problems in some of the localities. So coming to the IPM in sunflower, as I told you in other crop, deep ploughing is summer ploughing is important to expose the various stages of the insect pests or pathogens to the birds or hot sun. And especially wherever cutworm is a problem, you can go for a rigid furrow method of sowing for management of the cutworm. And the intercropping of sunflower with pigeon pea, especially short duration pigeon pea, and determinate type at 2:1 ratio effective against foliage feeders. And the intercropping groundnut sunflower with the cotton also lowers the thrips and gasset infestation. And a clean cultivation, especially destruction of the Parthenium, it is the alternative host for this necrosis diseases. That is very, very important, which will prevent which multiplies there, and through vector it will transmit to the sunflower crop. So growing your border crops like maize, sorghum, pearlmint, check the movement of thrips. That is also one of the important one. Then mechanical control, like as I told you, in other crop collection destruction, egg mosses, early installed larvae or damaged leaves, that will be effective. Especially for parakeet management, you can use these polythene bags. That is a traditional one, or you can use the latest reflective ribbons, like a bird scurry ribbon, like it's commonly available in market, like polypropylene metallic shiny ribbon with a red or label on one side and a silver white on other side. This is the one. This you can use for 4 to 8 meters between two ribbons. This is common, traditionally used by the manual humming or noise making devices also available that can be used for preventing damage by the parakeets. Especially in here, one important thing, we should not install any bird purchase, because here the several parrots and parakeets they are causing damage. You should not install any bird purchase, especially in sunflower and safflower crop. You should not recommend. And there are several botanical and biological control options are available. Addition of organic amendments like castor cake or neem cake, especially 200 to 400 kg per hectare, or Trichoderma harsianum, 2.5 to 5 kg per hectare, along with the farm amendment also recommended. Similarly, neem seed kernel extract or any neem formulation can be effectively used to prevent the egg laying by the leptopteran pest, and also effective on sucking pest. And similarly, neem seed kernel powder can be used 10 gram per liter, especially during the bud formation stage, that will repel the birds. Garlic bulb extract also, neem leaf extract also record, reduces the leaf blight diseases intensity. As many NPV can be used for management of sporometer or helicorp or even BT can be used at the rate of gram per liter. Coinciding the early install RV of leptopton pest. Similarly, there are several chemicals reported based on ACRP trials like spinosaurid or chlorantraniliprole effective on leptopton pest like semilupar, sporoptera, hairy caterpillars, and helicorpa. Similarly, seed treatment with imidacloprid thiamethops are effective against leafhopper and thrips. And sunflower have 4 insecticides formulations registered for management of some of the sucking pests like acid and whitefly and thrips, imidacloprid and thiamethoxam, seed treatment and foliar spray. They are having label claim. Similarly, some of the fungicides like metalaxyl for management of downy mildew, either through seed treatment or through foliar spray, to say having label claim. Similarly, one biopesticide, Regularma viridis, vegetable powder also registered at the rate of 6 gram per kg seed for management of seed rot and rot diseases. And also you can use through soil application up to 2.2 to 2.5 kg along with the farmyard manure to prevent these diseases. This is all about fungicides having label claim for sunflower diseases. So actually in the Google Play Store we have oilseed anti-glands and all the IPM practices and pest management practices are available in that internet. You can use of this mobile app for management of the pest. Especially these oilseed crops are eco-friendly. Actually, there are several natural enemies and pollinators are visiting. So during flowering stage, better to avoid any broad spectrum insecticide to conserve the predators, parasitoids, and pollinators. Thank you very much. This is all about IPM in oilseed crops. If any questions, kindly please. Hello, sir. Sir, am I audible? Hello. Sir, am I audible?
Yes, sir.
Yes, sir.
Any questions, sir, please?
Hello,
Sir.
dear participant, any question? If there's no question, we thank Dr. Durai Murgam for very elaborative and good lecture. Sir, is really wonderful to hear you once again. I hope the things will be benefited to our participants, and these because these participants are from the world, all part of the country. So really thankful for sparing the time. I hope next time also when we request you to share your experiences.
Sure,
Thank you
sir.
very much, sir. Thanks a lot.
Thank you, sir, for your opportunity and also the participants. Thank you, sir. Thank you for the opportunity. Thank you, sir.
Okay. So, friends, now we'll have your session at valedictory section will be at 2 o'clock, and the chief guest will be Dr. VP Chahal, ADG Extension ICR. Will be starting at 2 sharp, and I request two of your people, 2-3 people to give your feedback. So please be ready for that. Otherwise, I have to take name and to tell you to give your feedback, but I don't want to do that. So please, you select yourself. Now, Dr. Rekha, what else? Over to you. So we'll hear from Dr. Prajapati also. Your feedback we will hear today, right?
Yes, sir.
Huh? Okay, Dr. Rekha had put already placed the feedback and the post course evaluation. She had kept already with you. Just go through quickly and please submit it. You will have 20 minutes to there. So please see that. I hope Dr. Rekha, I'll request to put this also in WhatsApp group. But you have to mail it, right? I hope it will come mail. Huh? Yeah. So I am requesting Dr. Rekha also, so to give you feedback and as well as in the post course evaluation also in that way. So I hope in chat box it is there. Please check it. Chat box. And WhatsApp. Okay, it is also done on the WhatsApp group. She told me like this also. So please see that. Uh, you please do needful quickly. So we'll meet at 1:45. 2 o'clock they will join, but we will meet at 2 o'clock. And I request all of you, please switch on your what videos also at that time. So Dr. Rekha is over. So please tell them, instruct them to give that. Somebody
Exactly.
writing he is not in WhatsApp group, so he's not in WhatsApp group. Please see that it will be given to them, right?
Yes, sir. Yes, sir.
So Dr. Rekha, please take care,
They
right?
have shared the link for feedback as well as post-course evaluation for the training program in the chat box. I'll share the both of them on the WhatsApp as well, on the WhatsApp group and on the mail also. I'll send to all the participants who have received the the link for the training program that participants have received. Kindly submit your post-course evaluation as well as your feedback form as soon as possible. Before valedictory, we need feedback forms. By 5 o'clock today, all the links will be closed, and after that, you cannot submit your responses, and we won't be able to issue the certificates. So pre-course evaluation is already completed, and we have closed the link. We have seen that some people have like twice and thrice they have sent the pre-course evaluation. So we will filter that out and find out how many no duplications are there. The same goes for feedback as well as post-course evaluation. Only fill your form once and fill it by 5 o'clock today, so that we can issue certificates on time. Is there any query related to that?
मैम एक क्वेरी है कि जो वैलिडेटरी फंक्शन है 2 बजे से कितने बजे तक रहेगा मैम?
Those say I think we'll finish by 3:15 maximum. 1 hour. We have kept it for 1 hour, but by 3:15 it should finish. I think.
ओके मैम, एक्चुअली क्या है, मुझे मेरा 4 बजे एक यूनिवर्सिटी में गेस्ट लेक्चर है, तो
By
एक्चुअली
that time it should. I think by that time.
एक्चुअली 3 बजे मेरे को एक्यूरेट 3 बजे निकलना पड़ेगा, इसीलिए मैं पूछ रहा हूँ मैम। एक्चुअली 3:35 तक मैं निकल जाऊंगा।
Kindly give your feedback before leaving.
यस मैम, इसीलिए मैं पूछ रहा हूँ। एक्चुअली मैं 3 बजे तक निकल जाऊंगा, इसीलिए पूछा मैम।
क्या हो गया प्रजापति जी? 3 बजे पूरा खत्म हो जाएगा।
ठीक है सर। एक्चुअली
ADG also have some meeting.
जी
So
जी
we are
जी।
trying level best. So those are giving feedback. Your participants are giving feedback. Give crisp feedback.
Yeah.
Only, right?
And I request again that please give your feedback. Otherwise, we have to call the name, and it does not look good at that time. So
Sir, I will start first.
Okay. So when we call for the feedback, please send your feedback forms as well, because we'll share the whatever stats we get from the feedback form, that will be also shared during the training validity session. And we hope that the training was good. Like that, we are hoping. Let's see what kind of feedback, and please give your feedback in such a way that if you find any scope for improvement, write down on the comments so that we can also improve further to make our training program more effective and more comprehensive and more deliverable. So that I think we can close for now, so that we can send all the feedback form and sir. Hello, Dr. Sagan. Okay. If there are no further queries and announcements from our end, also we don't have any announcements. Sir, should we close for now, so that participant can fill all the forms? No,
I didn't get what you said.
Participants have to fill all the forms, so we can close for now.
Yeah.
By 1:45 we'll join back, sir.
Yeah. Okay. 1:45. Yeah.
1:45 we'll join back. So
if they fill up the feedback, we have a chance to quickly analyze and to tell, right?
Yes. Kindly, we are closing for now so that you can fill all your forms on time. And by 1:45, we'll join back. I'm sharing the link. We have already shared the link in the chat box, and we are sharing the link on the WhatsApp as well as on the mail. It's we are breaking for now. Thank you.
Respected Chief Guest, ADG Agriculture Extension, Dr. VP Chahal sir, and Honorable Director officiating Dr. Mukesh Segal sir, who himself is a training coordinator and training director, Dr. Mahadesh Shiru sir, and all other resource from our beloved resource persons who have joined with us for your today's valedictory program, and my dear participants. And welcome to today's valedictory program. Today we are here in the last chapter of this training program, which was organized for 5 days from May 9 to 13 on integrated pest management for sustainable agriculture. Now I take a pleasure to welcome all one and all for today's valedictory program. First of all, I would like to welcome our Honorable Assistant Director General Agriculture Extension, Dr. V.P. Chahal sir from Indian Council of Agriculture Research. Even after having his busy schedule, in fact, sir is supposed to attend one important meeting immediately after this valedictory program, but still he has accepted our invitation, and he is here to grace us. He is here to address us. On behalf of ICAR-NRIIPM and Manage Hyderabad, we wholeheartedly welcome you, sir, for today's valedictory session. I would like to welcome Dr. Mukesh Saigal, Course Director and Director officiating ICAR-NRIIPM to be a part of valedictory program. Welcome you, sir. I would like to welcome all other resource persons, honorable resource persons who has delivered their lectures as per our request. Welcome all the speakers, all the resource persons. I also welcome Mahadesh Shiru sir, who is a training coordinator, joined from Manage, and he who is a collaborator for us to organize this training program. Welcome you, sir. Not the least, I welcome our beloved participants, without whom our training program could not be happened. I welcome you all. And now I would like to request Dr. Rekha Balodi, training co-coordinator, scientist from ICAR-NCAPM, to give the training report, please. Dr. Rekha, please.
Thank you, Dr. Raghavan. A very good afternoon to all the dignitaries and participants who are present in this valedictory session of 5-day training program on integrated pest management for sustainable agriculture. This is a collaborative training program between Manage and ICAR-NCIPM, in which we started the training program on 9th May with the inaugural session. The inaugural session was chaired by Dr. S.C. Dubey sir, ADG Plant Protection and Biosafety, ICR New Delhi, and guest of honor was Dr. DG sir, DG Manage from Hyderabad, and Dr. S.C. Dubey sir on that day he flagged the issue of ground level implementation of IPM, and how we have to initiate the applications, how we have to collaborate and take it to the ground level, the practical problems that we face in the integrated pest management implementation at the ground level, they should be taken care of. And the same issue was even flagged, and it was suggested and highlighted by our honorable DG sir from DG Manage sir also, that who told that extension and technology should go hand in hand. Technology development alone and extension alone cannot do the job, and both things together should be taken care of. And he even together they told that in this 5-day training program we should be able to sensitize and do the capacity building of all the participants who have joined us from the different parts of the country. So for like updation of their knowledge in IPM, so that the same can be transferred to the farmers in the farmers field level, sir. And we on the same day we had 4 sessions. The first session was delivered by Dr. TVK Singh sir, in which he discussed about the past, present, and future of the IPM. and how, taking consideration of the present scenario, we have to take up the future problems and take care of them. And then about the IPM, general IPM, Dr. Mukesh Sehgal sir told about the general IPM on that the same day. Then we proceeded with the IPM in cotton by Dr. YG Prasad sir, and IPM in pulses by Dr. Konandram sir. And the day ended with the session on the project formulation and project formulation related to IPM. In that session, we discussed about how to go about in the project formulation related to IPM, about the agribusiness incubation facilities which are available with the ICR, and how the IPM can be inputs, and IPM can be an opportunity for the entrepreneurship development related to the agripreneurship and agriculture. Second day, we started with the role of microbial biopesticides by a very renowned scientist, Dr. H.B. Singh sir. He told about the importance of microbial biopesticides, their registration issues, how these microbials should be taken to the field, and what are the field application and field opportunities farmers have for using the microbial biopesticides at the field level, followed by the use of ICT tools and the case studies of use of ICT in implementation and dissemination of IPM was taken by Dr. Mukesh Sehgal sir. He talked elaborately on the ICT tools, and he studied, he gave the case studies where we have ICAR-NRIIPM has successfully used the ICT technology in disseminating the information related to IPM to the farmers and implementation of IPM at the farm level. Then they discussed about the use of nanotechnology related to the pesticide development and their use in IPM by Dr. Shakil sir, who is principal scientist in IRI. And he also talked about how the nanotechnology can be instrumental in pesticide development related to microbial biopesticide and safer pesticides for IPM, so that the menace of the chemical pesticide can be reduced and their judicious use can be implemented at the ground level. Then IPM gadgets, safer IPM gadgets and IPM tools were discussed by Dr. S.K. Singh sir, principal scientist ICAR-NRIIPM, who talked about how these gadgets can be a safer alternative to the chemical pesticide and can supplement the use of microbials and botanicals and biopesticides for reducing the use of chemical pesticide, as well as effective pest management, so that we have better yield and better cost benefit, better benefits to the farmers. And then we talked about the invasive pests. Then Dr. Krishna Kumar sir, ex-DDG horticulture, we talked about invasive pests and their menace and how those pests should be managed, and the precautions should be taken by the country for managing those invasive pests, and as in IPM, how we should take care and what strategy should be implemented. Third day, sir, we started with a very important topic related to protected cultivation by principal scientist from ICAR-NRIIPM, Dr. Satyendra Singh sir. He elaborately talked about the problems faced in specifically in the protected cultivation related to the pests, related to nematodes as well as diseases, and how we had what could be the management strategies specific to the protected cultivation. That session was followed by a very important session which is good agricultural practices related to the post-harvest integrated pest management of the crop produce, which was dealt with by Dr. Revan Siddha from IIPR Kanpur. He talked elaborately about how chemical-free pest management can be done in the post-harvest produce. Then the session was followed by a panel discussion in which we had an interaction between the participants and various experts which were called from the different parts of the country related to entomology, pathology, Dr. Dinesh Singh from IRI, Dr. N.A. Shakil sir from IRI, from BAU Sabor, Dr. Aditya. From Northeast also we had many experts who discussed elaborately about IPM, their constraints, the problems at present that at the farmers will that are faced, and the participants also interacted with the with the our experts, and they talked about the recent issues and the problems they face in the field conditions. The session was followed by a very important session on role of pollinators in IPM. We understand that the role of pollinator is very important for not only for honeybee production but increasing the quality yield and as well as environmental protection and biodiversity conservation. So that the lecture was dealt by Dr. R.K. Thakur sir, and he talked elaborately about the pollinator conservation, honeybees conservation, and their rearing. Then next day was fourth day. We had a session, a very elaborate session from three eminent scientists from NBAIR, Dr. Richa Vashne, Dr. Ankita Gupta, and Dr. Gracie, in which they talked about elaborately about the predators, parasitoids, and biological control per se, and DNA barcoding. And various issues they are flagged by them, that how the predators and parasitoids should be used, what could be the better opportunities, and what could be the ideal condition in which the predators and parasitoids should be used. The session was followed with the session on IPM in vegetable crops by Dr. M.N. Bhatt sir from ICAR-NRIIPM. He talked about the vegetable ecosystem and how it is important. It is very important to judiciously use pesticides, chemical pesticide in vegetable ecosystem, so that the vegetable remains safe and we have a low residual amount chemical pesticide in the vegetables. For that, Dr. Aman Bhatt sir elaborately talked about the eco-friendly pest management strategies in vegetables. The session was followed with a case study of ESAP by Dr. Prabhu Raj sir, in which he talked about the electronic pest surveillance and how the ICT again can be very instrumental in disseminating IPM. And that session was followed by a session with our coordinator from Manage, Dr. Mahantesh sir. He talked about the role of Manage and how the various collaborative programs are developed with the Manage, and how Manage can be very, very instrumental for disseminating agricultural knowledge to all parts of the country. And then Dr. Sehgal talked about the IPM in cereal crops, which is very important because in cereals also the residue issue, the pesticide residue issue is very important. Therefore, the integrated pest management in cereals is very important. It was dealt with elaborately with Dr. Sehgal. Then today we had two sessions in the morning. First was IPM in sugarcane by Dr. Shamila Roy ma'am. She talked about the pest management in sugarcane, elaborately covering all the diseases and pest problems. And the session last session was IPM in oilseed crop, which was dealt by Dr. Durai Murugan sir. And in this training program, sir, we have more than 350 participants who have registered, and around 120 participants are always present, and they interacted with all the experts which were called from various parts of the country. The participants really interacted well, and they raised very relevant questions which are very which they found problems they found in the field conditions. Further, we have shared the feedback form with them, sir, and we have received feedback from many participants, which I would like to share with you all. Give me a minute, sir. I hope my screen is visible. So this is the event feedback that we have received from all the participants. Around 116 participants have given their feedback, and overall, what we can see that maximum participants have given the overall session-wise feedback to be all the sessions to be in the excellent category, and almost all the sessions they have given highest number of participants have given the excellent feedback for session-wise. Also, 67% participants feel that the session content was either excellent, or 46% rest of them feel that it was very good. Like that, they feel, and overall feedback of the training is also very encouraging. Though we have flagged somewhere we have fair, one or two participants have said the fair. So we will see, sir, what issues they have faced, whether they found at what level we have seek the suggestions from them. So we will see where we can improve, so that we can further improve the efficiency of our training program. And many participants they say that they would like to take up another program like this, and the content of the program which was covered during these 5 days, they found it very excellent, and like that they are telling. And further they have received some uh, suggestions from the participants. This is the feedback that we have from the participants. So the feedback that we receive from the participants is encouraging, sir, but we'll still look forward for listening from the participants that how they felt during the training program and how we can improve for further. For that, I'll request Dr. Raghavan to please proceed asking the participants. Thank you, sir, for giving me the opportunity for presenting the training report.
Thank you, madam. Thank you for your extensive report on training program. Now, before proceeding, I would request all our dear participants, at least at the valedictory, please switch on your videos so that we can take some photographs for our documentation. I request the participants to please switch on their videos. Okay, moving further. Now I would call upon Dr. Mahantesh Shiru sir, training coordinator, to give highlights some key issues on furtherance in IPM extensions.
Thank you, Dr. Raghavendra. At the outset, I'm very thankful to Dr. V.P. Chahal sir, Honorable Assistant Director General Agriculture Extension, for joining this valedictory session of important training program on integrated pest management. And on the same note, I must also thank Dr. Mukesh Sehgal, Dr. Raghavendra, Dr. Rekha, and all the faculty at NCIPM for delivering this program so effectively. And the feedback and the response of the candidates is an indication that this training program was worth attending. And it's also heartening to note that some of the participants, just in a glance, I could sense that they want a physical program of similar kind of a training program, focusing on the same content. So probably that's a very good sign, and also a indication for all of us to think about a physical program. And sensing the sentiments, with so many online training programs happening, and people attending to their office works besides attending through these training programs, it's not that easy. But holding on to these 100 and plus participants was very, very encouraging. And I think probably the resource persons must have come thoroughly prepared, both for delivering the content in a fitting way and also making the content relevant for the needs of majority of the participants. And coming to the issue of IPM itself, I think it has been deliberated enough in the last couple of days. But most importantly, what is happening is, this IPM is just like an important tool, and it should be in the hands of every stakeholder associated with agriculture. Starting from the industry, starting from the input dealers, the farmers, the academia, and all these people have a role to play in order to further the IPM practices in agriculture. And more importantly, in the wake of sustainable development issues, not affecting the food security and the nutrition security issues, the IPM has to be pursued in a scientific way. And the spirit of science must also be respected in ensuring the profitability to the farmers and also the safety of the consumers. There are diverse experiences drawn from across the world, and these experiences should be examined in the context of Indian farming situation. And we have so many challenges, especially in among the small and marginal farmers, where majority of our farmers belong to the small and marginal holding farmers. How far the IPM practices can be made adoptable by these farmers is an important issue. And for this, science must not only focus on the core issue of technology itself, but the technology dissemination and adoption must find solutions as what makes the technology penetrate among the stakeholders. So this is an important issue that, as an extension scientist, that I always feel must be on the priority of the scientist involved in developing the technology on IPM. The second most important responsibility on extension personnel itself is to understand the IPM in totality. Because most of the times what happens, we see any picture with a smaller perspective and we lose the hindsight of having a larger picture behind the development of the technology itself. So therefore, an extension scientist must obviously be very thorough, and he cannot have an excuse that no, I am not belonging to this discipline. And in a worst case, what an agricultural extension officer must be able to do is connect to a right kind of resource person who is an authority or an expert in the integrated pest management. So these are all some of the issues that we must be looking for. And as I said on the first day of the program, to manage joining hands with NCIPM or any similar institute for furthering these technologies to the stakeholders in the agriculture and agriculture discipline, extension discipline, is very, very important, because the partnership is the way to move forward, best way to move forward. Because now if I see just, if I go by the names and the images of the participants, so what I feel is probably many of the faces and the names are quite new to us. That also gives me a assurance that we are able to reach different sections of people. And this is what the partnership, this is what the partnership brings to the table. And as I also mentioned, the input dealers and the agripreneurs also have a role to play. Most importantly, the technological interventions through the startup entrepreneurs in the form of developing potential IPM tools, their dissemination, making them cost effective at the time of commercialization. All these are very, very important and critical issues if IPM has to really find place among the regular practitioners of agriculture. And I think this is what that comes to my mind immediately. And probably the keener look at the feedback will help us to design a similar program or a better program based on the feedback. And sometimes Dr. Rekha was mentioning about some of the fair comments or something like that, but they are entitled to their opinions. But what I can feel and what I can say confidently is that the program delivered with the collaboration of Manage and NCIPM certainly is very best. And I think we will again see what can be done to address the issues raised by many of the participants. With this, I thank all the team led by Dr. Mukesh Sahgal sir for delivering this important training program. And we will stay in touch for future collaborative training programs. And again, I thank Dr. V.P. Chahal, Honorable ADG sir, for gracing this occasion to be with our participants. Thank you one and all. And on the same note, I must also thank participants for wonderful participation. And as I said, fatigueness is one factor. The pressure from the office table is another factor. But yet, you have managed very skillfully to attend this training program, and I'm sure there are many more lessons that you could have drawn, and you will certainly make use of these important lessons in your day-to-day life. So with this, I thank the organizers for having given me this opportunity to share my opinions. Thank you one and all.
Thank you very much, sir. As you rightly said, IPM should be conceived in a very scientific way from both the scientific community and the stakeholders, the grassroots stakeholders, especially the extension functionaries. And in this regard, sir, we, the ICAR-NRIIPM and other crop-based ICAR institutes is working hard to enhance the capacity of grassroots level extension functionaries in various programs through capacity buildings, through seminars, through workshops, likewise. And regarding physical mode of training program, certainly, sir, if time permits, in due course of time, by grace of God, if everything settles well, this COVID endemic gets subtled, then we look forward to manage to come ahead to have a collaboration with ourselves and to organize such a physical programs in due course of time, sir. Thank you very much, sir. Now it's a time for our participants to give their feedbacks. Now I request our beloved participants, anyone among you or a few participants, please come forward to share your feedbacks to us so that which can help us to enhance ourselves to for the betterment of the training programs in due course of time. Participants, please.
Good afternoon, sir. Good afternoon, sir.
Deepak, sir, please.
Yes, sir. Sir, I am Dr. Deepak from KVK Udaipur, Rajasthan. First of all, thank you to manage and ICAR-NRIIPM for really, this was really practical. Am I audible, sir?
Yes, sir, please. Sir.
Yes.
Yes, really, this training was really a practical and knowledgeable training. Many topics have been covered in this training, 5 days training, especially general IPM, IPM modules, pulses, ICT tools in IPM, followed by a case study by Dr. Mukesh Sehgal. It was really a good knowledgeable topic, and besides this, the other topics really very practical and knowledgeable. So I am in the behalf of KVK, it's really thankful to manage and NCIPM. Really, this training we learn more things, especially about biocontrol, microbial pesticides, Pest Regulation Acts, predator parasite, and recent advances in biological control. Dr. H.B. Singh, we learned also from him about the trichoderma, many species, microbial pesticides, and IPM in cereals. Especially this is the topic by Dr. Sehgal sir, because there is a residual problem in cereals. This was really a remarkable topics, and especially also Dr. Surendra Singh about the pheromone trap, light trap. We learn, all the participants, we learn many things from this training, and we ensure that the things we learn from this training, we will apply it in our field and try to make the environment safe and go for IPM. IPM. Thank you, sir.
Thank you, Dr. Deepak. And any other participants who would like to share their views?
Hello, sir.
Yes,
I am audible.
Yes, please. Yes, please.
Yeah. I am Shoikat Majumdar from Meghalaya, assistant professor, University of Science and Technology. First of all, I want to extend my heartiest warm regards to NCPI and manage ICAR. Just last 5 days, we had a excellent and wonderful productive session through various separate sessions, specialized session. Whenever we used to go to field for our, basically I'm from extension background, agriculture extension. So whenever we used to visit the field, interior field, the basic question we used to come to our front that regarding IPM. So what's their controlling method, what's the management, what's the practices. So through these lessons, sir, we got a vast knowledge, theoretical as well as practical. Thank you so much. Looking forward to more like this. Thank you.
Thank you, Dr. Majumdar.
Thank you, sir.
Very good afternoon, sir.
Neeraj ji, please.
Myself Neeraj Kumar Prajapati, or sir, जो ये 5 दिन की ट्रेनिंग रही है, इसमें IPM का एक पूरा पैकेज एंड प्रैक्टिस पूरा कंप्लीट सॉल्यूशन मिला है। जो अभी तक हम लोग के साथ एक इश्यू आता था कि हम लोग को कहीं कहीं पे थोड़ा थोड़ा एक नॉलेज मिलता था, और एक IPM का सबसे बड़ा इश्यू यही रहता था कि हमें एक चीज के बारे में पता रहता था, किसान के खेत में कोई दूसरा इंसेक्ट आ जाता था तो वहां पे हम लोग को फेल हो जाते थे IPM सजेस्ट करने में। तो ये जो पूरे 5 दिन की ट्रेनिंग चली, इसमें पूरा कंप्लीट स्टार्टिंग से लेकर एंड सॉल्यूशन तक, और हर एक क्रॉप वाइज और पर्टिकुलर इंसेक्ट को भी लेके उसका बायोलॉजिकल स्टडी बोला जाए, या फिर उनका पूरा एक मतलब कोई भी इंसेक्ट के ऊपर, या फिर उसका कोई भी प्रेडेटर, मतलब पूरा एक कंप्लीट स्टडी का इंफॉर्मेशन मिला। अब ये जो काम है ये NCIPM ने अपनी तरफ से तो कर दिया है ट्रेनिंग देके, लेकिन इसको सक्सेस करना बड़ी बात है, वो हम लोग के ऊपर है। जो आज इसमें हम 120 पार्टिसिपेंट्स हैं, आज सिर्फ एक सर्टिफिकेट लेके और इसको एक हर जगह शो करते हुए घूमे कि NCIPM से हमने ट्रेनिंग किया, वो हमारी सक्सेस नहीं है। हमारी सक्सेस ये है कि जो आज हम 100% यहाँ से निकलेंगे, जो हमने नॉलेज लिया उसको प्रैक्टिकली एक्सपीरियंस करें, और जो हमारा व्हाट्सएप ग्रुप है उसको मेंटेन रखें, और हर एक की प्रॉब्लम उसमें हम पोस्ट करते रहें, तब जाके हमारी ये ट्रेनिंग पूरी सक्सेस होगी। और एक बार दोबारा मैं NCIPM और मैनेज परिवार का बहुत बहुत धन्यवाद करना चाहता हूँ। जी सर।
धन्यवाद नीरज जी। Anybody else?
Good afternoon, sir.
Yes, sir. Yes.
Prashant Nalurkar from RSBC Jhansi.
Yes, sir. Yes.
First of all, I'll thank to Dr. Mukesh Sehgal, Shuru sir, and his team to organize this IPM training. It helps us to refresh many things, which will help us to implement it in future in field. My feedback is that I found that there was a bit bias in the content towards management, IPM practices towards insect pest management. There was less content for the plant pathological diseases. So I'll request in future, if we organize such kind of training, there should be balanced content of the because IPM goes equally for pathological as well as insect pest. So it will be good. Otherwise, most of the lectures were very good, and they has helped us to refresh and enrich our knowledge. Thank you.
Thank you, Jambulkar sir. Your concern is been taken care of, and it will be dealt in due course of time. Next participant.
Hello, sir. Can I say something, sir?
Yes. Please introduce yourself.
Hi, Raghavendra. This is Kuldeep Singh Jadon from Kajri, Jodhpur.
Yes.
Yeah, I would like to compliment you that you people managed this training very well for 5 days.
Right, sir.
And one good compliment is that you people managed all the lecture in time. Not a single lecture crossed limit of 5 p.m. Because this is a scorching heat in Jodhpur, and staying in places like it's very difficult here. But for this training, lectures, interesting lectures were there, and they refreshed my memory for implementing the IPM in field and interaction with farmers. And one peculiar thing that I noticed in this training program, that role of ICTs.
Right.
There are around 3 or 4 lectures on ICTs, e-surveillance, e-SAP, and then Sehgal sir's lecture. They were very good, and maybe we can also develop something here for this arid zone. So something is there, then we will be happy to collaborate with you people. And thank you very much. Thank you very much for this training, and thank you very much in NIIPM and Manage. Thank you very much.
To your information, sir, we have a very strong base. We have a very strong team of ICT working here over here in our NCIPM. So certainly, sir, in near future, we would we would wish to work with other crop-based institutes, not only with the ICT related, in any other issues which related to pest management. We are always welcome for all our stakeholders, sir. Thank you. And participants, next participant.
Good afternoon, sir.
Sir, please introduce.
Myself, Dr. A. Mohammed from NRCB Banana.
Yes, sir.
Actually, I was working previously Lab Research Institute. Recently joined in Banana. That's why I want to update my knowledge in IPM. Immediately this training has come, immediately I have proposed to attend the training. But exactly I have updated. Actually, after 10 years, I was working some production aspect. The pest management just I have started. I have proposed a project also eco-friendly pest management. Because of this purpose only, I want to refresh my knowledge. But very beginning lecture, present, past, future IPM like that it has started. Then brilliant in ICT. ICT tools, I want aware about ICT tools. ICT tools and ESAP. Still our ICR system we are following, it is very good. This ICT tools, it will go to individual crop wise. Some linkage may be created with among the ICR Institute. Then we can take help from all the people. Then one tool, then all the farmers, especially in specific crop wise, in a specific region wise, if you will go means it will be very good. Like network project mode, then ICT tools. Remaining thing, I think new knowledge we can learn, but ICT tools are very specific one. ESAP ICT tools. For this, please, I am my recommendation also, but in future we can do some work on ICT tools in network mode. Thank you.
Sure, Dr. Mohan Sundaram sir. Your concern is taken care of, and we'll work hard in this line, sir. Thank you. Next, any participants would like to share their views quickly?
Good afternoon, sir. I'm Dr. Shiva Joyce Rosen from Tamil Nadu Agriculture University. First
Yes,
of all, I
ma'am.
want to congratulate both NCIPM and Manage for conducting such a useful program. The best part of it was that time you could able to start by 10 and finish by 4:30 or 5. So it was very excellent in time managing. And many of the things are also new to me, like the successful ICT like ESAP and wonderful lecture on poly. Then about the quarantine pest, what Krishna sir had taken, and Sir Mukesh sir also had given a very good overview of all the things. And one more thing that is to be appreciated is we have covered all the crops invariably, the field crops and also the horticulture crops. One thing I want to add is we can just come up with something on the insecticides also. So since you are talking about IPM, let us also know about the different things that happens in the with the insecticides also, sir. Otherwise, it was a very excellent program, very useful, timely, sir. Thank you.
Thank you, madam. Thank you, dear participants. Keeping constraints of time in view, we'll stop the feedback session here and we'll proceed further. For rest of the feedbacks, anyhow, we have received all the feedbacks individually from your end, so we'll take care of. Next, I will request our Honorable Director Officiating and Course Director and Local Coordinator, Dr. Mukesh Segal sir, to give his director remarks, sir. Dr. Mukesh Segal sir, please.
Thank you, Dr. Raghuvinder. Esteemed Dr. Chahal, ADG Extension ICR, Dr. Sarur from Manage, Dr. Raghuvinder, Dr. Rekha, other faculty of NCIPM and Manage, as well as my dear participants. It is very, very pleased to note that majority of you are feeling satisfied what effort we are making or what we had made. This is a really, you can see the whole training program I can see is like an IPM itself. You have seen that there is a rescheduling of the lecture depending upon need. Likewise, we have to make our insecticide or our pest management strategy, we have to reschedule according to the pest. So that is very, very important. And second, somebody told timing. That is very, very important. The most important is monitoring. Monitoring is very, very important. So likewise, you can see we start from 10, right? We have started 9:45. Every time we have a recap also. And we try to finish up by 4:30 or 5. And so it shows that it is a strong monitoring we are doing. In the field also, we are so disciplined. Every day we tell our farm, just like in many times I told, or many times my colleagues have told you in this during this training program, a regular monitoring is important. And therefore, role of ICT play a very important role, because if the farmer have the problem today, you cannot tell him that after 1 week you do this. The thing is gone. So you have to tell only on time. So therefore, you have to be on time always. So what we do, we have a 12 WhatsApp group at the moment. We are working throughout the country. Almost, I told you, 21 states we are covering. And I wish that you are now also our IPM ambassadors. Somebody told that just like we have to now transmit to the farmer. That's definitely. I really tell you, with the help of KVKs and extension worker, NCIPM had reached more than 1.5 lakh farmers. We are reached. They know IPM. Means they simply don't and not listen. They know how to do the IPM also. Majority of them, and majority of them, when we withdraw, they still continue. You can see a classical case we have in many states, like we have in Uttar Pradesh, 2 3 places, Madhya Pradesh, you have Rajasthan, that's like Udaipur, you have the Tripura, you can have Meghalaya. Means wherever you go, Karnataka, wherever you go, you feel that farmers are practicing IPM. They have definitely constraint. We are just like in the beginning, I also told you we are requesting our director to have more entrepreneurship programs now. So I hope in coming days we may in this 5 year plan we plan that 500 entrepreneurs will be trained. So likewise we are training. No doubt we are training the farmers. We have the cooperative societies developed by NCIPM in Assam. You can they are selling their product through ICT only. During COVID, Assam lemon, Khasi mandarin was sold in whole Assam only on their app. We have developed the app, and that app they have used up. And then we also have the FPOs. We have IPM marts. So if I request all of you, those have attended very seriously. I saw many of you have attended seriously. Therefore, you know the feedback also. And so if every one of you develop 5 villages every year, and if you need some help from us, we are ready technically, literature wise, whatever the feedback, whatever the need you have. Sometimes we have critical inputs also can be given, but in very limited resources we have. But whatever we have, we can extend. We can, those are working in tribal area, northeast, they know that they are working with me and they are working excellent. So I wish that the same knowledge and whatever constraint you feel that, please, this WhatsApp group what we have is filled up, will be continue. But please say don't use good morning, no wishes. We want you you just tell your problem. Let's we have a solution. We interact with us. We are developing e-platform. Just like we are in that e-platform, NCIPM making effort, every agency will come together, just like, and then one will pose a problem and other will also give a solution. That includes farmer, pesticide dealer, state agriculture department, SAUs, KVKs, ICAR Institute, and NGOs, pesticide dealer, every or critical inputs, all will be there. So this will be our e-platform for us, all of us, means between you and us, there is a e-platform. Please, please bank upon us, and we will be ready to help you out. And we are very thankful to manage. First, I tell you that this is my in my career. I started the such type of program in 1995, 11 October 1995. Until now, we have 73 program we have conducted. But this, and always we have a customized, means one particular state, two one Atari, two Ataris, like that. All Ataris we have covered up. So like that, we are developing state, means Maharashtra. We are going for Gujarat. We are going for Tripura. But we never have such a versatile group. Meaning, always we have the some extension worker group. Either we have extension worker group, scientists, SMS, farmers. We have already organized more than 5,000 farmer field school at NCIPM, and every year we are doing more than 200. And in COVID also we have conducted 112 farmer field school online with the help of our KVKs, extension worker, and others. So this is but we thankful to manage. So to brought us such a big platform, I said this is a big platform. At least 400 people, 402 people have registered. They know IPM. They know what is course content, what IPM includes. That makes at least 20% our job done. Means a person know what IPM includes. Then you are all joined here and interacted. I am feeling very happy. Somebody say attendance. I say attendance is not required because screenshots are there, people are interacting, people are doing a good job. That is sufficient for us. That is motivating to us, and we made our efforts. You are seeing, it is not that NCIPM only faculty is transmitting to you. Those are successful, those are pioneers, just like Professor TBK Singh. You have the Prabhuraj, Dr. Prabhuraj is from Raichur. You have the Dr. HB Singh is from there. Dr. Sharmila Roy, means many people you can say from all over the country. We try to brought at a one platform. Those are, you can say, they are talking on our wavelength, same wavelength we are talking about. It means we have Dr. Durai Murgam, we have the UK Kondaram. They people are working on the same. Somebody told is very correct. Prajapati is telling very correct that sometimes people are not aware what IPM includes. And I'm very thankful to Dr. Rekha and Dr. Raghavinder. They have told you that how to develop an IPM project. It's very difficult because normally when we talk IPM project, somebody very correct. I know that somebody right only concentrate on insect pest management. But might have seen that when any person developing from NCIPM, we try to include from socio-economic survey, soil health card. We talk about the, you can say, pest, insect, disease, nematodes, weeds, everything we try to, and then economic analysis. I also shown to you how to do that. So that project formulation is also done it by these two people, and the most to the house I tell you, is very very satisfactory for me. Yeah, in this long journey of the human resource development, that this time we have hold panel discussion, and my time was short. I had an expert. You're surprised. I had an expert of 37, but we could have the view only of 13 in 1 hour, 1.5 hours. We try to we try to do that, but we have to sacrifice the lecture of Dr. Thakur. But that I cannot do because Dr. R.K. Thakur, you have seen Honeybee lecture, you cannot hear other than this. So therefore, the whatever we did, you keep in mind, it is not to pass a time. It is only to, we want you to make more knowledgeful, more ambassador, more trained, and I hope in future also you collaborate with us and we do a more successful IPM. I hope in our dream is 2050 every farmer trained with IPM. I hope you all can make with us. Thank you all. Thank you, Dr.
Thank you very much, sir. As our director, sir, and course the coordinators are emphasizing all our beloved participants to be IPM ambassadors. I'm pretty sure, sir, the participants on behalf of our ICR may now act as an ICM ambassadors to designate IPM to the grassroots level, especially to help the farmers to reduce as much as chemical consumption in our Indian agriculture. Thank you, sir. Moving ahead, going further, now I would request our Honorable Chief Guest, ADG Agriculture Extension, Dr. VP Chahal sir, to give his valedictory address. ADG sir, please.
Thank you. For the director of NCIPM, Dr. Mukesh Sagar, and he's also host director of this training program. Dr. Mahantes from Manage, the training coordinator, Dr. Raghavendra, Dr. Rekha Barodi, and all the participants of this training program and all those who have connected. It is my proud privilege to be with you during this valedictory session. I am very much thankful to NCIPM for giving me this opportunity and talking to you this afternoon. So thank you very much, Dr. Saigal and his team. Friends, as you know, as I have listened to all the stakeholders of this training program, my first and foremost view is that this training program has been conducted very professionally. Very professionally. I have seen the training schedule which you had given me day before yesterday. That right from the inaugural session to this validity session, you have planned a very good program talking out covering all the components of under the topic IPM for sustainable agriculture. All relevant topics have been covered, whether it is related to the overall brief about the past, present, and future of IPM, or crop production technologies, IPM strategies in vegetable crops, cash crops, or ICT in IPM, supported by many participants, post-harvest, gap for post-harvest, and protected cultivation, role of predator and parasitoids. All topics were briefed by Dr. Rekha. So my first and foremost reaction on this program is that it has been completed very professionally, and the most important part is that it shows the feedback of participants shows that the participants might have participated very actively in this 5-day training program. So I congratulate the organizers as well as the participants of this training program for their actively and professionally conducting and actively participating in this training. No doubt, the IPM is an important component of sustainable agriculture, and it is very timely that such type of training programs are being organized by our NCIPM and other institutes also. So, there is a need to develop some IPM models based on which a farmer can decide whether he should go for IPM or he should not go for IPM. So some models should be developed, which should include that what are the sources of information, how the IPM will be used by the farmers, what are the timings for application of the IPM, and so some models should be, I think, developed by NCIPM and other research organizations as well as the training organizations, so that we can help farmers to decide. So some decision support systems or like that, those models should be there. So this will help farmers to apply the IPMs in the field. So along with the, I think when we develop a IPM program, then I think we should also make integral part its evaluation of the IPM part. Evaluation should be the integral part of the IPM program. Sometimes we do the summative evaluation, but I think formative evaluation is most important, because if some deficiencies are there in our IPM program, while we are working with farmers, then we can make some midway corrections in our approach, and we can deliver it more effectively. And there is also there is a need for adopting long-term approach, not short-term and site-specific approach. Now the time has come that we have to shift our IPM approach from short-term research for short-term preventive and curative measures to long-term strategy. So such type of research as well as extension programs should be developed, which can be of which can have long-term consequences. Because adoption of IPM technologies requires the long-term decisions, not just short-term decisions. If a farmer has adopted in one season and next year he is not adopting, so there may be some reasons why that farmer is not adopting. So we have to develop some long-term strategies for that purpose. Then I think there is a need for developing good agricultural practices and standards for IPM for different crops, whether horticulture, whether vegetables, fruits, field crops, or other crops. So that is also required. And while we develop the IPM program, then sometimes we think that this is better for farmers. But the farmers do not realize that he has a need for adopting the IPM strategy. Farmer does not realize. So we have to first identify the needs. Need assessment is very much important, and need assessment not of farmers, of all the stakeholders, whether farmers, whether NGO people, whether input dealers, whether government organizations or public representatives. Involvement of public representatives is important because they have a role in policy making, and if they are convinced, then they will help in developing some policies in favor of the use of the IPM technologies and best practices. So their consultation of all stakeholders for studying the needs and the overall requirements for adopting the, and I have forgotten the consumers. Now consumers are becoming more and more sensitive, and they are demanding that residual free farm products should be made available to them in cities as well as in villages. So it is our duty to consult all before taking up the IPM strategies. And IPM produce of IPM field, IPM adopted field, the produce should be marked some differently. Some we have to make some arrangement so that farmer should get some premium price if he has or she has adopted some IPM strategy and IPM practices in production of his produce, whether vegetable, fruit, or other field crops. So that should be that level of to his crop, or level to a village, or level to some specific fields or some village or some specified area. So that I think that can be thought of, and we can discuss on that matter, and it is important. I think there is a need to develop a IPM matrix. On one side, we should have some IPM impact areas, whether environmental impact of IPM is there, or health impact is there, or economic impact is there. So we have to develop that so that our IPM program is more comprehensive, inclusive, and on the another hand, the IPM focus areas, impact areas, which are the impact areas, whether IPM focus areas, whether we have given our focus on various production systems, whether plant-based production systems or animal-based production systems, because there is a very close relationship between the soil, plant, animal, and human beings. So we have to understand all the concerned who are concerned with the soil. If soil is not properly dealt with, then plants will be produced with some residues, and if residues plants with residues will be fed to animals, then animals will be affected, and ultimately the human beings are affected. So such things have to be taken care of. So we have to, in a way, we have to make our IPM programs smart programs. It should be specific so that we are on the track while implementing this program. Its outcomes should be measurable and attainable. The objectives of that IPM program, whether training or whatever, should be attainable and result oriented and time specific. Specific time frame should be there. So in a way, our program should be smart. Then I think there is a still the IPM has come of IPM strategies have come of age in our country also, right from the time when the farmers field schools were started. But still there are number of barriers, and there are many constraints at many various levels in the adoption of IPM technologies and best practices by farmers, whether these are related to the technologies, whether these are related to the funding of the IPM programs, or the overall awareness among the farming community, the social marketing aspect is lacking, or we are also lacking in educational approach. It is not a input distribution program. The IPM program is a knowledge intensive program, and it is always a educational program. So we have to give much focus on educational part. If we are able to educate a person, then he will, if he has learned the principles behind the IPM technologies and best practices, if he has learned, then he will certainly apply. So our most focus should be on educational approach rather than input distribution. So though the trainings and the demonstrations and provision of critical inputs, all these are very important, but our most focus should be on the educational approach. Then sometimes people do not understand the agro ecosystem of the area and the knowledge and entry behavior of the farmers, knowledge level of the farmers, training needs of the farmers, and what are their requirements, and how much and which pesticides they are using, and what are the different things which can be modified or which can be adopted by the farmers. We do not understand. So understanding of the agro ecosystem in a whole, in a wholesome approach should be there. So the understanding of agro ecosystem is most important while we planning any IPM activity in the area. Sometimes people feel that IPM technologies and best practices are very complex and not easy to adopt and all that. Such type of perception is there in the field. So that can be taken care of. How to, so farmers, if we take the farmers perspective while planning our program, then such type of perceptions can be taken care of, that IPM technologies are complex and cannot be taken. Then sometimes our farmers feel that while adopting the IPM technologies, they are taking risk. They feel that they are taking risk, and they do not trust the overall the IPM practices. Sometimes it is there in the field, and input dealers and all that, they have their profit motive, and they do not advise the farmers to go through and adopt the IPM use of limited use of chemicals and most effective control measures of the crops. So that aspect is lacking among the input dealers and those providers, because they have their profit motive. And lack of convincing information is also one of the constraints. So we have to develop a full proof communication strategy with our IPM program, so that everybody is convinced that what we are saying is trustworthy, and people, and there should be clarity. And while we are demonstrating the technologies, the complexity perceived by farmers should be simplified, so that and we should be able to demonstrate the relative advantage of the technology, whether in terms of economics, or whether in terms of its usability, or whether in terms of its environmentally safe or residual free, or all that. All those issues should be clearly mentioned in our communication strategy in clear terms. There should not be any confusion. In my opinion, our communication strategy is most important. So because the success of any program depends upon depends upon clearing the doubts, and simplifying the things, and understanding the overall perspective of our farmers users. So all these things are should be taken care of. And we should also focus on the regular capacity building programs. You have taken a right step for conducting this, but continuous capacity building is most important, and the capacity building of extension personnel is the most important. Because the farmers on the one hand, the scientists and KVK subject matter specialists, and the extension personnel are on the other hand. So capacity building of all these three, farmers, subject matter specialists, as well as the extension personnel. Because the scientists and the subject matter specialists of KVKs are very limited, and they cannot reach to each and every farmer. So we have to develop the capacity of extension personnel. So the technology flow, we should have a very smooth technology flow, develop a technology flow plan, so that suppose NCIPM has some technology, that technology has to pass on to KVKs. The KVKs have to include, do the capacity building of extension personnel in the field, and the extension personnel should have the capability to convince the farmers to adopt the technologies. So that technology flow should be clear, and long-term strategy should be there, I think. Then I think government is giving focus on organic farming for sustainable agriculture in future also. So there is a scope for plant-based biopesticides, bioscience, and all those, the organic farming also. So some specific programs should be organized for organic farming, because the availability of input is a serious constraint. Organic inputs are not available in the field, and there is a dearth of availability of standardized and very effective organic inputs. So on those issues, I think we should have a separate program for that purpose. Then I think we should also give some our attention on future of IPM. What is the future? And there should be a clear-cut communication strategy for spelling out the future of IPM. So that is very much essential. Then I think there is a need for institutionalization of the participatory approach of IPM process in all crops, and for sustainable agriculture. So this is institutionalization of IPM approach and participatory approach, and the participatory technology program, participatory technology development. Right, when we conceive any research project, then farmers' perspective should be taken care of, and we should involve farmers whether while conducting some surveys, the farmers should be involved, or whether we are monitoring some pests in the field, so farmers' involvement is important. Whether we are collecting some data related to IPM, then farmers' involvement is must. So in all these activities, there is a need for ensuring the farmers' participation, so that they can own the program. Unless and until farmers own a program and they feel that they are important partner in the overall application of IPM technology in the field, then there are chances that the program will be successful. And already you have discussed that you have developed a WhatsApp group and all that. So I think networking is very important. Networking of farmers, networking of scientists, networking of KVK SMS, networking of the extension personnel also. So networking, already Dr. Shagal has given focus on this issue, and so I would like to develop some long-term research-based. You might have already started working on that long-term research programs on IPM, and there is a need for developing ecologically based novel IPM technologies, so that we are safe. Environment is safe, human beings are safe, animals are also safe, and the natural resources are also managed properly. So all these things are very important. So once again, I would like to thank Dr. Mukesh Sehgal and ICIPM and Manes for giving me this opportunity to be with you during this very interactive session. I congratulate team ICIPM, team Manage, and congratulate all the participants of this training program. And it is expected that everybody will take some action. So after attending a training program is one thing, getting a certificate is one thing, but after that, taking action is more important. So you have to work with the farmers and their organizations, whether they are farmers producers organizations, farmer producer companies, or self-help groups of farmers and their other organizations or NGOs. Some you might have developed a framework in your mind that I will do this, I will do that. But that should be, that framework should result in some concrete actions on your part. So thank you very much again.
Thank you very much, sir, for your valuable chief guest address, valedictory address. As you have flagged upon the very important issues like IPM model, region-specific IPM model development, decision-making supports, need-based need assessment of stakeholders, IPM certification of labeling of IPM producers, requirement of IPM matrix development. Likewise, certainly, sir, this has given a way for us to think of it and do successful tremendous research and extension activities in the areas which has been highlighted from your end, sir. Thank you very much, sir, for gracing us, for sparing your valuable time with us. And now I would request Dr. Rekha Balodi to deliver a formal vote of thanks. Dr. Rekha, please.
Thank you, Dr. Raghavan. It is often said that the end is the start of the new beginning, and we believe that the end of this training program will be a new beginning for dissemination of IPM throughout the country. believing that the participants from all the parts of the country have gained a lot of knowledge and they have gained a base for further enhancing the effectiveness of the IPM at the grassroots level. It is my proud privilege and honor to deliver the formal vote of thanks for this very important training program. We extend our heartfelt gratitude and thanks to ADG sir for gracing the occasion and like motivating us and blessing us with his words. Sir, the issues flagged by you, highlighted by you, starting from the model development to the metric development and the constraint analysis, need assessment, they are they are the guiding milestones for us, and they will be the guiding principles for further making or development of the research programs, extension program, and capacity building program, as well as for the improvement of the existing program which we have developed till now. Sir. We'll take care of all the issues flagged by you, and we assure you that we will work in the framework that you have given us for the further development. Sir. Thank you so much, sir, for gracing the occasion. We extend our heartfelt thanks to DG Manage, Dr. Chandrasekhara, and the coordinator from Manage, Dr. Mahantesh Shirul, sir, for giving us the opportunity for conducting this training program in collaboration with Manage. It was indeed a privilege, as Dr. Mukesh Sehgal sir said, that we have been conducting the capacity building program for the Ataris, but they are mostly location specific, and we used to cater the SMS of one or two areas, not the whole country. But because of this collaboration with Manage, we were able to increase our reach, and we could manage to gather the attention from all the parts of the country and gather many people who belong, not only scientists, extension personnel, research scholars, and farmers, to bring them all to one platform. And many of the participants, they have remain actively actively participating in the training program. We believe that with this collaboration we have actually increased the efficiency of NCIPM as well as reach of the NCIPM to extend the training program, and we further believe that we will be able to develop further many collaborations with Manage in development and conduction of such training programs, including the physical training programs if the time and opportunity come across us. I further thank Dr. Mukesh Sehgal sir, Director Officiating and in charge HRD, Course Director and Coordinator for this training program. Sir has been instrumental, motivational for development of these training program and capacity building program. He has always encouraged the youngsters in these training program, and in a way we are also he is doing capacity building for us also, that we are learning so much through these training programs, and we are able to communicate with the higher, we have increased our reach, and we are learning so much in the process, sir. So we thank you, sir, for your motivation and inspiration, and we also promise you that we will always try to come up to the expectations that you put on us. We further thank our dear participants. It was we believe, we know that it was a tireless journey for you guys, because we actually tried to make the whole IPM in one capsule and deliver it in a nutshell. We actually bombarded all the information to you, but thanks to your attention and all the concentration and all the interaction that you gave us from day 1 to day 5th, all the sessions were really interactive, and we believe that we were able to deliver the content to you. And at the same time, as Dr. Mukesh Sehgal sir said, we request you and we believe that you will be the IPM ambassadors to the country, for reaching the different parts of the country and to reaching the different farmers' needs. Thank you so much, dear participants. And we again request you to please join us for the further training programs, whichever is organized by ICAR-NRIIPM. I further thank all the seniors from NCIPM who have helped us in conducting this training program. I thank all the resource persons who have been really instrumental in delivering the content of the training, as well as providing us the lecture notes, which are being compiled and they will be given to all the participants for their reference in future. We thank all the administrative and financial staff from NCIPM as well as from Manage, who helped us in conducting this training program very smoothly. I thank one and all who are present here, and I believe that with this training program, we further increase our collaboration, our networking, and we will be able to deliver to the cause of IPM, and we will be able to deliver to the cause of safe food security, a healthy nation, and a happy nation. Thank you so much. Jai Hind.
Thank you very much, ma'am, for delivering a vote of thanks. With the permission of Director sir and Shirur sir, we would like to close the program, and there is a announcement regarding the certificates. The certificates and after completion of the post training formalities, the certificates and the resource materials will be sent to the participants through their emails which they have provided during their registration. Thank you very much. Thank you very much. ADG sir, namaste sir. Thank you very much, sir.
One last thing, please complete your feedbacks and post-course evaluation by 5. The links will be closed after that. Thank you.
Okay. Thank you, Dr. Rekha, Dr. Raghavinder, and all. Dr. Chahal, Dr. Sirur. Thank you very much. We meet again once again. Thank you.
Thank you, sir. Saurabh, please close the meeting.
Thank you, sir. Thank you.
Thank you,
so
Dr.
much.